Good afternoon, faculty members and PG students. Today, webinar on molecular, genetic, and clinical aspects of cancer. I welcome you all on this webinar where we are going to deal about different aspects of cancer, which is always a disease which is always dread about and which is a second most common cause of death after cardiovascular disease. It is said like we are going to have uh, all the PG students present on different aspects of this cancer. So introduction will be done by Ms. Soni Limbu. Differences between normal and cancer cells will be done by Rachana Puri. Types of cancer will be done by Dr. Richa Karki. Etiology of carcinogenesis will be done by Dr. Sabita Nepal. Molecular biology of cancer cells will be done by Dr. Manoj Acharya. Cancer stem cells and immunology will be done by Dr. Sneha Sinha. Role of growth factors and their receptors in cancer will be presented by Ms. Sapna Subedi. Cancer and metabolism will be done by Mr. Bisal Khatri. Role of oxidative stress in carcinogenesis will be done by Dr. Piyush Gupta. Tumor markers will be presented by Dr. Roshan Kumar Thapa. And lastly, treatment will be done by Dr. Dilip Kumar Yadav. So without further wasting our time, let's go ahead with the presentation. So I'd like to call upon the first presenter for this session of webinar. I'll stop sharing and you can share soon.
Only of those that are epigenetically regulated, not mutationally regulated. And avoiding immune destruction, cancer cells avoid immune destructions by B and T cells by paralyzing the immunosuppressive agents or factors such like PDL1 or NK cells. Enable replicative immortality. Cancer cells have limitless dividing potential and tumor promoting inflammations. Cancer cells promote inflammations through mainly two mechanisms, through intrinsic factors and extrinsic factors. Extrinsic factors such as mediators and inflammatory cells, and for intrinsic factors and polymorphic microbiomes. Microbiomes and polymorphic microbiomes and polymorphic microbes have a very important, significant role in causing cancer. And these microbes can cause cancer through like several mechanisms, such as microbes itself can cause cancer through the process called carcinogenesis, or through different therapeutic agents, or through cancer outcomes. And activation, invasion, and metastasis. These hallmarks describes that cancer cell they spread, or they move from their primary site and living and spreading to the other parts of the body. And this metastasis can happen, or it has a various stages, such as first of all they are locally invasive. They form the tumors, then they enter to the bloodstream and the lymphatic system. They travel through the bloodstream and lymphatic system, then they escape and they go to the distant metastasize, and they form the tumor cells and they grow there. And inducing or assessing vasculature. What this hallmark describes is that cancer cells is rapidly dividing and it requires more energy. And this process is described by the Warburg effect. And senescent cells, they are cells that has completely stopped dividing due to stress, and these senescent cells can act as a tumor suppressors or tumor activators. And genome instability and mutations. Genome instability mainly means is there is dysregulations in the process of the cell cycle, such as mitosis. During the mitosis process, there is dysregulations of that process, and because of that, and that results in the mutations. And resisting cell death. This is one of the hallmark of cancer, where the cancer cells they avoid dying even though this body tries to kill them by evading the process called apoptosis, by activating different pathways like p53 pathways, and deregulating cellular metabolism. And another is unlocking phenotypic plasticity. It disrupts in unlocking phenotypic plasticity. This hallmarks describes that the cancer cells disrupts the cellular differentiations, and through different mechanisms like de-differentiations, trans differentiations, or blocking differentiations. So I like to thank you. Thank you. Now I would like to welcome Rachna for the next topic.
So here is the content. I'll be talking about the introduction, what is cell, and the difference between the normal cell and the cancer cell. At first, as we all know that cell is a structural, functional, and the biological unit of all the living organisms. And these normal cells act as the body's basic building blocks. As the cell consists of the different organelles within it, and each organelle has its one specific characters, which helps to maintain the correct functioning of the tissues, organs, and our whole body. Some characteristics of the normal cell are so normal cells control their growth by using the external signals. That means they only grow and divide according to our body's needs. And the normal cells undergo the program cell death, which is called the apoptosis, as a part of our normal development. This is because to maintain the tissue homeostasis and in response to the unrepairable damage. Normal cells stick with each other with the help of the selective additions that is produced by themselves. And another is they differentiate into the specialized cells with the specific functions. So there are some basic differences which helps to differentiate the normal cell with that of the cancer cells. First one is the cell cycle. Normal cell usually consists of about 18 to 24 hours to complete one complete cycle. And this cell division is usually regulated within a balance between the dividing cells and the resting cells. Whereas in the cancer cell, though the cell cycle time is similar with the normal cell, but the percentage of the cells in the dividing phase is increased by 2 to 5 times. And this uncontrolled proliferation is mainly due to the deregulation of the checkpoints. Checkpoints in the G1 to S phase, which is called the start or restriction checkpoints. Another is at the G2 or M, in between the Z2 and M phase, which is called the major checkpoints. And when the cells enter into the Z0 phase, which is called the resting phase, they usually stops dividing. But in cancer cells, what happens is they skip this Z0 phase and they continuously divides and they forms the large in number. Another is the cell proliferation. The proliferation coefficient is the ratio of the cells in the dividing phase to the ones in the resting phase. And in normal cells, growth is limited and this is regulated according to our body's needs. But in what happens in tumor cell is they grow very rapidly. That is, they have the high proliferation rate and they divide and their doubling time is ranges from 10 to 450 days. For example, if a cell has the doubling time of the 100 days, then they takes about 10 to 8 to 10 years to form the size of 1 cm. Another differential characteristics is morphological changes. The metabolic cells are morphologically different from the normal cells. Normal cells becomes malignant after undergoing into the transformation. Here we can see in the figure, normal cell comes in the many shape and sizes. They have the regular structure and they have only one nucleus and very large cytoplasm, and they consist of only one nucleolus, and they have the fine chromatin as well, and they grow in a monolayer pattern. Whereas also we can see in figure right, which is cancer cell, they have the large irregular nucleus, and their nuclei is also prominent, and they forms the multi-layer structure. And the nuclear to cytoplasmic ratio of the cancer cell is also very high, that is in 1:1 ratio, whereas the normal cells nuclear to cytoplasmic ratio is 1:4 to 1:6. Another characteristics is the contact inhibition. Normal cell usually divides when they comes in contact with the another cells, and this helps to ensure them into a organized growth due to the contact inhibition nature. Whereas cancer cell lost this contact inhibition nature and leads to the uncontrolled growth and the overcrowding of the cells within the same place. Another is the tissue adherence. Normal cells adhere to the other cells and the tissue with the help of the protein called the vinculin. Vinculin is a cytoplasmic actin binding protein, and here we can see in the figure left, in the presence of the vinculin, the E-cadherin, which is another transmembrane glycoprotein, that is stable and expressed at a cell surface, and they strongly form the adhesion between the cells. But in the figure right, we can see in the absence of the vinculin, there is the loss of cell adhesion, and they disassemble, and this E-adherin is also lost when the vinculin is not active. So in what happens in cancer cell is, due to the malignant transformation, this vinculin gets inactive because the phosphorylation of the tyrosine kinase, and this inactivation of the vinculin decreases the adhesion ability of the cancer cells and results into the metastasis. Another is the role of the sialysin. Usually the charge within the cell, the surface charge of the cell is in the balanced form. So the normal cell exhibit a balanced surface charges due to the presence of the sialic acids. But in cancer cells, the sialic acid, which is also called the N-acetylmuraminic acid, the concentration of this sialic acid is very high, which leads to the increase in the negativity charge of the cancer cells. And this presence of negative charge on the surface of cancer cells prevent their adhesion between the cells, and this leads to the malignancy and into metastasis nature. Another is the metabolic alterations. The significant alterations in the metabolic pathway occur as a normal cell get transferred into the cancer cell. Our normal cells use the aerobic respiration efficiently for the energy, but the cancer cell metabolizes the sugars mainly by the anaerobic glycolysis. By utilizing the role of enzymes of the citric acid cycle and the mitochondrial electron transport chain, which is called the Warburg effect, and the production of several enzymes and proteins can also be altered in the cancer cells. Another is the immortal nature of the cancer cell. The normal cell undergo the apoptosis, which is a programmed cell death, after a set of the number of divisions. But in cancer cells, avoid this apoptosis nature, and they, due to the mutation in the gene like P53, and they becomes the immortal. Also, a cancer cell activate the enzyme called telomerase, which maintains the telomerase length and allow them to divide indefinitely and become the immortal. Another is the metastasis nature. Normal cells are localized and do not invade the other tissues due to the presence of the adhesion molecules. But the cancer cells releases the enzyme like the collagenase and the stromycin, which help them to pass through the membrane barriers, leading to the migration of the cell. And migration usually takes place via the blood vessels or the lymphatic system, and a significant decrease in important addition molecules like E-cadherin facilitates their migration. And the cancer cells also have the increased activity of the matrix metalloproteinases, and these enzymes help in the migration of cells by degrading the basement membrane proteins. Now, last one is the angiogenesis. Normal cell growth is dependent on the pre-existing blood supply, but the tumor cells secrete the angiogenic substances like vascular endothelial growth factor, and this VEGF acts through the tyrosine kinase receptor, which is present on the membrane of the endothelial cell of the blood vessels or lymphatic systems, and which stimulates the multiplication of the endothelial cells via the specific mechanisms. And this VEGF is upregulated by the hypoxia inducible factor 1, which is a potent stimulant for the new angiogenesis, and whose concentration is increases during the hypoxia condition. So here is my references. Thank you. Now I would like to call another presenter, Dr. Richa Karki.
Everyone, respected faculty members, seniors, and myself, Dr. Richa Karki, is it on a topic, type of cancer. Before going to the types of cancer, I am going to briefly tell about different types of cell growth. In different types of cell growth, hypertrophy. In hypertrophy, there is increase in number of increase in that causes increase in size of organ. There is increase in number of cells due to certain stimulus. Increase in numbers, whereas in reversible series, in which one cell type is changed into another cell type, whereas dysplasia is irreversible change in which one differentiated cell type is replaced by another cell type, and dysplasia can be further changed into neoplasia and neoplasia and its type. I'm going to tell about it briefly in my introduction. Neoplasia means new growth, and the collection of new growth and collection of composing new growth. Neoplasia is defined as a genetic disorder of cell growth that is triggered by the acquired or less commonly inherited mutations affecting a single cell and its clonal progeny. Tumor originally describes swelling neoplasia. All tissues are composed of two components: neoplastic cells, with that constitute the tumor parenchyma, and next is stroma, made up of connective tissue, blood vessel, and cells of adaptive and innate immune system. In types of tumor, we have three types of tumor: benign tumors, malignant tumors, and mixed tumors. Benign tumors at the site of the adhesion and generally inamiable to the surgical removal. Naming of benign tumors of mesenchymal cells usually to be simple in general. The topic stroma is added to the name of cell type from the of fibroblast cells are called as fibroma. Cells of cartilage tissues are called as chondroma, and so on, adenoma and cystadenoma. Next is malignant tumors can invade and destroy the existing to the distant metastasis and a distant site or metastasis. Malignant tumors are collectively referred to as the cancer derived from the Latin word that have different added to any part and that disease on it and absence manner. For example, malignant tumors, sarcomas, lymphomas, leukemia, and carcinoma. That I will talk briefly in later slide. And mixed cell tumors. Mixed tumors are all of these elements arise from a single nucleotide clone capable of producing both epithelial cells and mesenchymal cells. For the classic example of mixed tumor, we can take a salivary gland, which contains epithelial components scattered within and nucleus, and that may contain island of cartilage or bone. Next is type of cancers. In types of cancer, there are more than for the organ or tissue where the cancer from. For example, lung cancer starts from the in lungs, brain cancer start in the brain. But I am dividing cancer on the base of type of cells that form them, such as epithelial cells or squamous cells. So the cancer are differentiated into 7 types: carcinoma, sarcoma, leukemia, lymphoma, multiple myeloma, melanoma, brain and spinal cord tumor, and other types of tumors, germs, endocrine, and carcinoid are included. First is carcinoma. Carcinoma is the most common type of cancer by the epithelial cells, which are the cells that cover inside and out its surface of our body. Carcinoma that begin in the different epithelial cells and specific is the cancer that forms in the epithelial cells that produce fluid or mucus. Type this type of epithelial cells are from called glandular tissues. Most cancer of breast, colon, and prostate are adenocarcinoma. Whereas basal cell carcinoma is the cancer that begins in the lower or basal layer of epidermis, which is the person's outer layer of skin. Next is squamous cell carcinoma, is the cancer that the epithelial cells lie just beneath the outer surface of skin. Squamous cell also line many other organs, including stomach, intestine, lungs, bladder, and kidney. Squamous cells looks like a flat, like fish scale with viewed under the microscope. Squamous cell carcinoma called as epidermoid carcinoma. Transition cell carcinoma is the cancer that forms in the type of epithelial tissue called urothelium. This tissue, which is made of many layers of epithelial cells that can get bigger and smaller, is found in the lining of the bladder, uterus, renal cell, and few other organs and cancers. Cancer of bladder, ureter, and kidneys are found with cell carcinoma. Next is sarcoma. Sarcomas start in the connective tissue, as these are the tissue of our body. Connective tissue includes bone, fibrous tissue that supports the organ. The most least common that carcinoma, and there are two types. One is bone carcinoma and next is soft tissue sarcoma. This make up less than 1 in every 100 cancers person diagnosed every year. And osteosarcoma is the most common cancer of the bone. Most common types of the soft tissue sarcoma are leiomyosarcoma, Kaposi sarcoma, liposarcoma, and so on. Next is leukemia. Leukemia are the cancer that begins in blood-forming tissue of the bone marrow, are called leukemias. These cancers do not form solid tumors. Instead, large number of abnormal blood cells, leukemia cells, or leukemic plastic cells build up in the blood and marrow, crowding out normal blood cells. The low level of normal blood cells can make it harder for the body to get oxygen to its tissues, control the bleeding or fight infection. These are the most common types of leukemia, which based on how quickly the disease takes this or clinic, or on the type of blood cells the cancer is from, lymphoblastic or myeloid. Acute form of leukemia grow quickly and chronic forms grow more slowly. Next is lymphoma is the cancer that that begins in the lymphocytes. Disease causing white cells that are part of immune system. In lymphoma, abnormal lymphocytes build in the lymph node and lymph vessel as well as in the other organ of the body. There are main two types of Hodgkin's lymphoma and non-Hodgkin's lymphoma. In Hodgkin's lymphoma, people usually abnormal lymphocytes that are called Witt-Stokes cells. These cells usually from B cells. Non-Hodgkin's lymphoma have large group of cancerous cells that the cancer can grow quickly or slowly and can form from the B cells or T cells. Next is multiple myeloma and melanoma. Multiple myeloma is a cancer that begins in plasma cells, another type of immune cells. Abnormal plasma cells called myeloma build up in the bone marrow and form tumors in the brain all through the body. Multiple myeloma is also called plasma cell myeloma or tumor cells. Next is melanoma, is a cancer that cells that form the melanoma cells. That specialized cells that make the melanoma. That is the color to its skin. Most melanomas form on the skin because melanomas can also form in other pigmented tissues such as eyes. Next is brain and spinal cord cancer. Cancer that start in the cells of the brain or spinal cord. The brain is the body that electric along the nerve fiber. The fibers run out of the brain and join together to make the spinal cord, which also takes message from the body to the brain. These tumors are based on the type of the cells in which they formed and where the tumor astrocytic tumors begins in the star cell brain cells called astrocytes, which helps in the keep the nerve cells healthy. Brain tumors can be benign, not cancerous, or malignant can be cancerous. Other type of tumors are germ cell tumors. Germ cell tumors are the type of tumors that begins in cells and give rise to the cell process. These tumors can occur almost anywhere in the body and can be either benign or malignant. Neuroendocrine tumors are the forms from the cells that release the hormones into the blood in response to the signals from the nervous system. May make higher than normal.
Good afternoon, everyone. Respected faculties member, my seniors, and my dear friend. Now I am going to.
Go ahead.
Okay. Now I am going to start with the etiology of the cancer. I am Dr. Sobita Nepal. First of all, etiology of the cancer cell. First of all, the predisposing factors. there are the different predisposing factors for the etiology of the cancer for the cancers to develop, and these are the age, hereditary, and the environmental factors. First of all, going through the age occurrence of the cancer is mainly associated with the older people, that is age more than the 50 years old, and also with the children's via the retinoblastoma tumor, neuroblastoma tumor, and the Wilms tumor, and etc. And hereditary conditions such as inherited high risk factor play an important part at lesion in some type of cancer. For example, in some families, many women develops the hereditary ovarian cancer and the breast cancer as a BRCA1 and the 2. And next is the environmental factor. In environmental factor, mainly the lifestyle modification is the one of the predisposing factor. And in the lifestyle modification, first one is the cigarette smoking. And in the cigarette smoking, aromatic hydrocarbons, nicotine, carbon monoxide, and the nitrogen dioxide present in the cigarette smoke are responsible for the developing the lung cancer. And next one is the tobacco chewing. And in tobacco chewing, oral cancer in human being have been listed to the tobacco chewing. And next is the alcohol consumption, and increase the chances of the oral, pharyngeal, and the esophageal, as well as the liver cancer. And in the dietary, certain fungus such as the aspergillosis flavicus, which grows on rice, wheat, and the other grains, are also responsible for to cause the cancer. And next is the occupational hazards, and exposure to the asbestos, benzene, etc., may predispose the human body to formation of various type of the cancer. And next one is the therapeutic drugs, and these are the environmental factor to predispose for the cancer. And next is the mutagens. Mutagens, any substance which by damaging the DNA causes the mutation is called a mutagen. And the substance which by enhancing the rate of mutation convert a normal cell to the cancerous cells are called the carcinogens. And all the carcinogens genic substance are the mutagenic. And mutagens can be divided into the physical, chemical, as well as the biological and the viral. And under the physical energy, there is a visible light, UV lights, and the X-ray and the gamma rays. Mostly the visible lights are non-carcinogenic. However, it can become a carcinogenic in the presence of the photosensitivity agent, which generate the reactive oxygen species. And UV lights, it is the important carcinogenic agent, and sunlight is it's the main source of the UV light radiation. And the X-ray and the gamma rays, they are also induce the mutation by the radiation energy with the formation of reactive oxygen species. And physical and radiation energy. Radiation energy can cause the mutation by damaging the DNA by following mechanism, causing the single stranded break in the DNA, causing double stranded breaks in the DNA, and the deletion of the purine or pyrimidine based DNA, and by formation of pyrimidine dimer and cross linkage of the DNA strand. Next is the chemical carcinogen. It is estimated that almost 80% of the human cancer are caused by the chemical carcinogen. In some of the sources of the human cancer causing are in from the agriculture sector. It is some pesticides, insecticides, and in from the industrial areas like aniline dyes and the asbestos, and from the foods alpha toxin, And from the cigarette smoke, I already told the benzopyrene, and from the polycycline aromatic hydrocarbons such as the chlorothianine and the benzopyrenes, and the azodides aromatic amines are the binathalamine and the 2-acetyl aminofluorine. And from the nitrosamines and the amides, dimethyl nitrosamines and the dimethyl nitrosamines, and the drugs such as the cyclophosphamide, busulfan, and the diacetylbesterol. Next is the action of the chemical carcinogen. Mainly the action of the chemical carcinogen, there are the two type of the initiator carcinogen. They are direct carcinogen as well as the indirect carcinogen. Indirect carcinogen, also known as the procarcinogen, and direct carcinogen, they are highly reactive and interact easily with the target molecule like DNA to produce the cancer, and they do not undergo any metabolic activation or modification to body to cause the cancer. For example, the metochlorothianamine and the b-propionolactone, and the indirect carcinogen are, these are not reactive and does not require prior metabolic activation to become the reactive, and between are called the prominal carcinogen. And both the direct and indirect carcinogen are electron deficient, and they are reactive electrophiles which interact with DNA to produce the mutation in DNA and ultimately results in the neoplastic transformation and development of the cancer occurs. And next is the stage of chemical carcinogen. Mainly there are the two stages. First of all, it is the initiation, and then it is gone to the promotion as well as the progression. And the first of all, initiation, there is a irreversible change in the genome resulting in one or the more mutation, and it will progress to the cell division and malignant, which is the promotion and the progression of the cancer cell, and transformation of the initiated cell to the progression. And there is a one chemical test in the carcinogenicity, which is known as the Ames test, or test for the mutagenicity, which was launched by the Bruce Ames, which is a professor of biochemistry and molecular biology at the University of the California in 1974. This Ames test is an essay and inexpensive way to measure the mutagenicity of chemical. And Ames test does not determine if chemical is carcinogenic, and rationale of this test is greater than 90% of all chemical mutagen have been shown to be carcinogenic in subsequent animal tests, and the bacterium used in this test is Salmonella typhimurium, and that causes a defective mutation gene, making it unable to synthesize the amino acid that is histidine from the integrate in its culture medium. And however, some types of mutation, including this one, can be reverse or back mutation with the gene regarding its function, and these reverse are able to grow on medium lacking the histidine. And this is the procedure showing the Ames test, and here the strain of Salmonella typhimurium not synthesize the histidine, and amino acid essential for growth is used, and this essential is used, and this is done in the solid agar growth medium, and there is the added of suspected mutagen, and the diffusion of added mutagen is there and incubated for a time and again. And if chemical is a mutagen If chemical use is a mutagen, then the many colonies are formed, and many are the histidine positive revert colonies. And if the chemical is not a mutagen, then far fewer colonies are formed, and these are mostly the histidine negative. And next is the biological carcinogens. Approximately 20% of the human cancer are caused by the viral genome, which could be either the DNA or the RNA. Some of the important DNA and RNA viruses causing the cancer in human beings are listed below, such as from the hepatitis B and the hepatitis C, which is a DNA genome and which cause the hepatocellular carcinoma, and the human papilloma virus 16 and the 18, which is a DNA genome and causes the cancer of the uterine cervix, and the human herpes virus type 1, which is a DNA and which causes the sarcoma, and Epstein-Barr virus, which is the DNA genome.
So I think I'm audible and my presentation slide is being shared.
Yes, you are audible and your slide is also shared. You can go ahead.
Thank you, ma'am, and good afternoon, respected faculty members, my colleagues, and my juniors. So today I'm going to continue the webinar on the important topic, that is molecular basis of cancer. So today I'm going to present about a brief on cell cycle abnormalities, the activity of the telomerase, and the most importantly the apoptosis part, and different types of gene that causes cancer, that is the proto-oncogenes, and different tumor suppressor genes like retinoblastoma gene, p53 gene, DNA repair genes, and also some roles of microRNA in the cancer, and brief overview with the cancer epigenetics and about the familial cancer syndromes. So I think this cell cycle has been already been told by Rachana. So briefly highlighting it. So a cell, when it needs to get divided, so it need to pass through the cell cycle. So there are different checkpoints. Actually, there are 3 checkpoints. So one is known as S entry, commitment point, or the checkpoint. So this checkpoint ensures the cell is ready for DNA replication or not. There is another one, that is the mitotic entry checkpoint. It confirms the cell is prepared for the mitosis or not. And the next one is the mitotic exit commitment point. So it ensures whether the mitosis is completed before the cell division or not. So not only this checkpoint, but there are different other factors like the proteins, the cyclins, and different enzymes like cyclin dependent kinase, which are responsible for looking after the cell, whether the cell needs to get altered or the cell needs to go or needs to pass the cell cycle or not. And not only that, when if the cell is get halted in between the cell cycle, then the cell is whether repaired, or the DNA of the cell is whether repaired, or it may undergo the apoptosis, that is the programmed cell death. So before moving on to the apoptosis, I'll highlight a little on about the telomerase activity and cancer. So this figure, it illustrates the role of telomerase. So in normal cells, what happens is that the telomeres get shortened with cell division and leads to aging. But here now in the cancer cell, what happens is that, so there is one complex that is called the cell complex. So it is a protein assembly actually, and that helps to protect the telomerase, and it prevents the DNA damage response. But its dysregulation leads to the, or may promote to the tumorigenesis. And here we can see here the next, the activity of this TERT. This means telomerase reverse transcriptase activity with telomerase RNA component, that is the TERC, and epigenetic alteration helps or favors the tumorigenesis, and various cancers may be caused through it, like melanoma, GI cancers, breast cancers, brain cancer, thyroid cancers, sarcomas, and etc. So what is apoptosis? So it is a programmed cell death. So actually it is apoptosis is caused by the activation of the caspases. So activation of the caspases is the hallmark of the apoptosis. So there is the DNA fragmentation and cell shrinkage and formation of apoptotic bodies which are cleared by phagocytis, and it is without the inflammation. And there are two major pathways for apoptosis, that is the intrinsic pathway and the extrinsic pathway. So here we can see that in the left side we can see the intrinsic apoptosis process, whereas in the right side the extrinsic apoptosis process. So the intrinsic apoptosis process, it is always activated by the cellular stress. So and there is the involvement of the mitochondria. So here we can see that this is the cellular stress, and mitochondria, it plays a key role here. So due to cellular stress, what happens is that there will be the holes formation in the mitochondrial membrane, that is facilitated by the proteins, that is BAX back and BAK back. So we can see the full form. I've listed it at the above of my slide. So once there is a hole in the mitochondria, so different factors like this cytochrome C, SMAC, DIABLO, SDR2, so different kinds of factors, they get released from the mitochondria, and it helps to build the structure called apoptosome. So here, so after so these factors, what it actually do is that it activates this procaspase 9, and there is the formation of apoptosome. This apoptosome, it activates the caspase 9. So in intrinsic apoptosis there is the activation of caspase 9. So this caspase 9, it activates the other effector caspases like 3, 6 and 7 and induce caspase dependent apoptosis. But there are other factors like IAP. So IAP, what it does is that it is a regulatory process to inhibit the apoptosis, but the proteins like SMAC, SDAR2, what it do is that it inhibits the IAPs and the apoptosis is initiated. But this is the caspase dependent apoptosis in the mitochondria. But there is another pathway also here. So without activating the caspases also, it can initiate apoptosis. So here we can see the proteins like SDAR2, AIF, and INDO-G. So these are the factors. It can cause the cell death directly by damaging the cytoskeleton and DNA. Now here in the right hand side we can see the extrinsic apoptosis. So it is like an external command to self-destruct the cell. So the special receptors, that is known as death receptors, so we can see here, receive death signals. For example, we can see this FAS, and it is bind to the ligand. It is also known as FAS ligand. What is then after, when it gets bind to the ligand, then there will be the activation of this disc. So this disc formation takes place, and this procaspase 8. So in the extrinsic apoptosis, this procaspase 8 is activated. Further that, it activates the caspase 8, and there will be the other effector caspases activation like 3, 6, and 7, causing to apoptosis. But this extrinsic apoptosis is also linked with the intrinsic apoptosis. Here what happens is that, so there will be the mitochondrial involvement after the activation of this caspase 8. So this TBID, so this means truncated BH3 intercating domain death agonist, it gets activated. This causes the activation or the holes in the mitochondria, and it links to the intrinsic pathway also. So here in the far most right, we can see this dependence receptor. So this dependence receptor, what it is that it ensures the cells that the cells only survives in the right environment. So here we can see that in the green portion, that is the ligand. So ligand is not bind with the receptor. So in absence of ligand, so it can activate the procaspase 9, and caspase 9 leads to the apoptosis. So in summary, so in intrinsic is triggered by cellular stress, and it involves mitochondria and activates caspase 9, whereas extrinsic, it is triggered by external signals that involves the death receptors and activates caspase 8. And there is the dependence receptor. It ensures the cell survival only in the presence of its ligand, otherwise it triggers apoptosis. So why cancer cells are anti-apoptotic? Because these cancer cells are regarded immortal because they have developed mechanism to inhibit the apoptosis, and there will be the disturbance between the pro and apoptotic system. So that is due to the cell death signals are turned off and the cell protection signals are turned too much over here. So the most commonly mutated gene in various cancers is P53, which causes upregulation of the anti-apoptotic factors while downregulating the pro-apoptotic factors. So the different genes they play a role in cancer developments are oncogenes, tumor suppressor genes, and DNA repair genes. So there are two factors that promotes the cancer, that is the by initiating the tumor, and another is by promoting the tumor. So the various radiations or carcinogens that causing genetic mutations, that is the DNA damage, can start the process of the cancer development, whereas the proliferation of the mutated cell without directly causing the mutations like chronic inflammation and growth factors, they are the tumor promoters. Now the oncogenes. So what are oncogenes? So they are the genes that is responsible for the formation of the cancer, and they are found by the mutation of the proto-oncogenes. And proto-oncogenes, actually they are normally present in the body, and approx 100 proto-oncogenes are known, which code for the cellular proteins which regulate normal cell growth, proliferation, differentiation, and survival. So these proto-oncogenes get transformed to oncogenes by undergoing mutations in one allele of each gene or promoter. So here we can see this is the flow chart. This proto-oncogene, it is transformed into the oncogenes, and the growth rate of the cells will be increased by rapid multiplication, and the tumor gets formed. Now, how these inactive proto-oncogenes activate into the oncogenes? So there are different processes. So first of all, there is the mutation. So here we can see the point mutation. So this is within the gene. So a small change in the gene itself can turn it into the oncogene that is producing the abnormal protein. The next one is the within the regulatory element. So a change in the regulatory region also can cause the gene to make too much of protein. The next one is the translocation. So here is the new promoter or the enhancer. So a piece of DNA is here. So this piece of DNA, it moves to a new location and putting the oncogene under a new control element, regulatory element, leading to too much of protein. The next one is the fusion gene. So here the two gene gets merged and it creates a new gene that produces a novel or excess protein, the new or the excess protein. The next one is the gene amplification. So the gene gets copied multiple times here, so leading to the excessive amount of the normal protein. So actually there are 5 classes of oncogene: class 1, 2, 3, 4, and 5. So we can see over here also. So this is the general table showing the genetic changes in the proto-oncogenes to their roles in cancer by showing how they disrupt normal cellular processes. So for example, cyclin D, the main process is the translocation and the amplification. The activation mechanism is, and the process involved is in the regulation of the cell cycle, and oncogene associated cancer are breast, liver, and esophageal cancer. The next one, so suppose this let us tell this BCL2. So the process involved is the activation process involved is the translocation, and it is the apoptosis regulator. And due to the defect of the, so due to the oncogene associated with this may lead to the follicular B-cell lymphoma. So here are different other cancers. So how the proto-oncogenes have their roles showing to disrupt the normal cellular processes. So I can share you this slide on the later in the later. So there are different tumor suppressor genes also, and actually these tumor suppressor genes, the normal function is to inhibit the cell proliferation, but absence of this inactivation of the inhibitor of tumor suppressor genes may lead to the cancers. So there are different types, that is the P53. So it is the most important one and also regarded as the guardian of genome, and it regulates the cell cycle arrest, DNA repair, and apoptosis. The next one is retinoblastoma gene. It controls the G1S checkpoint in the cell cycle. The next is the BRCA1 and BRCA2 gene. It is involved in DNA repair, that is particularly the double strand breaks. P10, so it regulates the cell survival and proliferation by inhibiting the PI3K AKT pathway, and APC, so it prevents uncontrolled cell growth by regulating beta catenin levels in the WNT signaling pathway. So moving on to the retinoblastoma gene. So actually this retinoblastoma protein, it controls the cell cycle that is moving past the G1 checkpoint. So actually this retinoblastoma protein, it is bind with this regulated transcription factor that is also called as E2F. So E2 transcription factor. So what happens here is that, so this RB protein, it prevents the activation of this E2F. So which stops the cell from entering the S phase, that is in the DNA replication. But now when there is the presence of the growth signals through the RAS pathway with the help of the CDK cyclin, so it is the ATP dependent process. So this causes the phosphorylation of this retinoblastoma gene. So after the phosphorylation, the E2F gets separated, and this separated E2F causes gene transcription, mRNA translation, and further initiates the DNA replication. Now next one is the P53 gene. So here this phosphorylated P53, what it does is that it activates the transcription of P21 gene. So this P53 gene, so when there is a DNA damage, so actually it activates the ATM, that is the ataxia telangiectasia mutated gene. So what it does is that this P53 is inbound with this MDM2 protein, and when this P53 gets phosphorylated, this P53 that can otherwise it is a suppressor gene, but when it gets phosphorylated, it initiates the transcription, and after transcription, there is the activity of P21, that is the CD kinase K inhibitor, gets activated. This inhibits the G1 CDK cyclin, and it cannot phosphorylate the retinoblastoma protein, and there will be the cell cycle arrest. So in the next, this P53 also can trigger the cell death directly if the damage is severe. So it can cause the apoptosis or the cell death if the damage is severe. So there are two other genes, that is BRCA1 and BRCA2 gene. So here we can see in the normal tissue has only one functional BRCA1 or BRCA2 gene, but the tumor tissue often has both BRCA1 and 2 gene mutated. So in the B here, we can see that this BRCA1, BRCA2, and BRAD51. So it repairs the DNA breaks. Then there will be the protection of the stalled replication forks that prevents the fork collapse. So then after that, there are different Fanconi proteins. This fixes the crosslinks, so which is crucial for repairing DNA interstrand crosslink and maintaining genomic stability or integrity. But what happens in the tumor cell is that, so there will be the mutation of BRCA1 and BRCA2, and there will be the collapse of the replication fork, and the DNA breaks. And this DNA uses the error-prone pathway for the repair, that is the single-strand repair and non-homologous end joining repair, that may cause the genomic aberrations or the chromosomal abnormalities, and it causes genomic instability, cell death, or cancer. Then comes the P10 gene. So in the cell, we can see this is the tyrosine kinase receptor over here. So when the different growth factors get binds, then what it does is that it activates this PI3 kinase, which converts this PIP2, that is phosphatidylinositol phosphate 2 to PIP3. So it is phosphorylated. It activates PDK1, which in turn activates AKT, and that AKT activates this mTOR, and this mTOR, what it does is that it causes protein synthesis and cell growth, cell cycle progression, proliferation, and survival or decrease apoptosis. But what this P10 does is that it inhibits this pathway by directly converting this PIP3 to PIP2. Then comes the role of microRNA. So in microRNA, so here the two roles has been shown over here. So one is the convention. So normally tumor suppressing microRNA, what it does is that it normally reduces the oncogenic messenger RNA, but if they lose the function, the oncogenic mRNA get increases and it leads to the formation of more oncogenic proteins and promoting tumor growth. And oncogenic microRNA, now what it does is that it normally suppresses the tumor suppressor genes, tumor suppressor messenger RNA, but this overactivation reduces the tumor suppressor proteins, further supporting the cancer development. Now in the buffering role, so under stress condition, this microRNA, it acts differently. So microRNA may inhibit this oncogenic messenger RNA, and it may act like a tumor suppressor. And in the next one, microRNA, it may inhibit the tumor suppressor messenger RNA, and it may act like a oncogenic. So this highlights the dual role of microRNA depending upon the conditions, that is like stress. Now this is the epigenetics and overview. So the different factors like DNA methylation, so that is adding small chemical groups, that is the methyl group to DNA, means DNA methylation. So the effect of this can turn the genes off, stopping it making the proteins. The next one is the histone modification. So changing the protein histones around which the DNA is wrapped is known as histone modification, and the effect of this can either turn the genes on or off by making the DNA more or less accessible. The next one is non-coding RNAs, that is the microRNAs, which I have already talked. So these the small RNA molecules that don't make the proteins, but it can control the gene expressions. So for example, long chain RNA or microRNA, and their effect is they can block genes from making proteins or have been turning them on. So here are the different syndromes. So this is the familial cancer syndromes due to mutation of different genes. So the mutation in the genes like BRCA1 and BRCA2 may predispose the cancer syndrome like breast and ovarian cancers, and the associated tumors are mainly the breast, ovarian, bowel, and prostate cancers. TP53, so it may cause the Lee-Fraumeni syndrome, that is the osteosarcoma, and the associated other tumors are the childhood sarcoma, brain leukemia, adenocortical carcinoma, and early onset breast carcinoma. So due to the mutation of PTEN gene, there the cancer syndrome is called Cowden syndrome, where there is the tumors associated are the breast, gastrointestinal, or thyroid cancer, and the another one is STK11 and LKB1. So the cancer syndrome is Peutz-Jeghers syndrome, and most over the tumors is associated with breast, GI, pancreatic, and ovarian tumor. And the ATM gene, that is the ataxia telangiectasia mutated gene. So the associated tumors will be non-Hodgkin's lymphoma, ovarian, breast cancers. So these are the different familial cancers, and these are my references. Thank you. So I want to call upon the next presenter for the presentation. Thank you so much.
Cell, they are the small subpopulation of tumor cells with capabilities of self-renewable, dedifferentiation, inherent chemo and therapy resistance, which ultimately result in tumor relapse that can lead to failure of conventional traditional therapies. Its characteristic, it is self-renewable in nature, a small number in cancer cell. It is chemo-resistant as well resistant to radiotherapy. It has a microenvironment interaction, high metastatic ability, and it has a capability of expression of cancer stem cell markers. Now coming to how it has been originated. So first of all, in normal stem cells, they are the undifferentiated cell. They have a self-renewal capacity, so can undergo mutation to form cancer stem cells. And normal progenitor cell, they are partially differentiated cell and have capacity to undergo mutation to form mutated progenitor. And the mature cells, which are fully differentiated, they have a lack of self-renewal capacity. So due to that, the mature cells getting converting into the cancer stem cell as it has a lack of self-renewal capacity. In the CSA cancer, found in multiple malignancies, including leukemia and breast cancer like breast, lung, colon, prostate, ovarian, brain cancer and melanoma. And the most common method that has been used to identify this cancer stem cell marker is FACS, that is fluorescence activated cell sorting, and its principle is based on that of the flow cytometer. So here these are the list of the markers and the cancer type involvement. CD133, which is a marker for cancer like brain, prostate, pancreas, melanoma, colon, lung, liver, and so on. CD44 is a marker for cancer, colon, breast, prostate, and pancreatic cancer. Similarly, ABC, that is ATP binding acid, G2 marker, which is for cancer, pancreatic, breast, lung, limb epithelial, brain, prostate, ovarian, retinoblastoma, and so on. ALDS is a marker for cancer, breast, lung, head, and neck. ABC B5, which is a marker for cancer, melanoma, and similarly CD90, which is a marker for T acute lymphoblastic leukemia, glioma, and liver. And regarding the second part, that is on cancer immune surveillance. In the year 1900, a scientist named Paul Ehrlich gave an idea that tumor cells can be recognized as a foreign substance and can be eliminated by the immune system. Similarly, in 1957, another scientist, Burnett and Thomas, coined the term immune surveillance, which implies that the normal function of immune system is to scan the body for the emerging malignant cell and to destroy them. And according to those concepts, the tumor arises only if cancer cells are able to escape the immune surveillance. And that can occur either by reducing the expression of tumor antigen or by an impairment in immune response to those cells. That's why the immune system is patrol for abnormal cells, often halting the malignant cell growth. And only those malignant cells escape the immune detections can be detected in the clinical tumors. So in the cells of immune system, we know there are two types of immunity, innate immunity and adaptive. This innate immunity, which is present since birth, it has a rapid response, whereas adaptive immunity, which is present during the lifetime, it has a slow response. In the innate immunity, we have cells like macrophages, dendritic cell, natural killer cell, neutrophil, eosinophil, basophils, and so on. And in case of adaptive immunity, we have a cells like T cell, and under T cell, CD4 plus T cell, that is helper cell, and CD8 plus T cell, that is cytotoxic T cell, and B cell, that is responsible for antibodies production. So let us first look at the innate immunity. In innate immune response, it is the first line of immune defense against tumor from less specific component of immune response, that is the innate immune system, which is present since birth. And this mechanism prevents spread of malignant diseases and are not specific to that particular tumor antigen, but can also recognize a broad characteristic of the tumor cells. And under the innate immune response against tumor, we have NK cell killing tumor, and second is macrophage production of anti-tumor cytokines. And this macrophage production of anti-tumor cytokines will include tumor necrosis factor and interferons. So let us first look at the NK cells. That is NK cell recognition of the tumor cell targets. This one is here in the left side, we have a figure that they are the cells having MSC1 class 1 molecules. And when they are transformed into the tumor cells, the transformation may reduce the expression of that MSC1 molecule. And once the expression of MSC1 molecule is reduced, it is going to increase the expression of the stress molecule. And the stress molecule, we have MICA and MICB. This MICA is for MHC1 chain-related protein A and MHC1 chain-related protein B. And their main function is that are recognized by the CAR and KIR on the NK cells. On the left side, we have CAR and KIR. CAR is a killer activation receptor and KIR is a killer inhibition receptor that is present on the NK cells. And this main function, another we have a cytokine with anti-tumor activity. And when this activated macrophages, in here we have a figure where activated macrophages is releasing tumor necrosis factor alpha and beta, and similarly interferon alpha, beta, and gamma. This tumor necrosis factor alpha and beta induces necrosis of the tumor cells, And when injected into the animal with tumor cells, they can stimulate the necrosis of the tumor cells. And besides that, this tumor necrosis factor alpha also inhibit angiogenesis. It is a process that in which the growth of new blood vessels by decreasing the blood flow to the tumor. And besides that, we have interferon alpha and beta and gamma. It also induces the expression of MSC1 molecule, making the tumor cell more susceptible to cytotoxic T lymphocyte killing. Another we have adaptive immune response. And adaptive immune response against tumor is include antibodies include the first here let us look. This is TSTA, that is tumor specific transplant antigen, which are bound to the antibodies. And antibodies are known to be generated against the certain specific antigen which are present on the malignant cell. The second, it includes the direct CTLS killing, that is cytotoxic T cell can sometimes kill the tumor by direct contact with CD8 T cell. The third adaptive immune response involves delay type hypersensitivity reaction, which involves thymine 1 cell recruiting and activating macrophages, which attack and kill the tumor cells. And another we have in the induction of T cell response to the tumors. So here let us look at the figure. We have two part categorized. The first is induction of anti-tumor T cell response, and second part effector phase of anti-tumor C T lymphocyte response. Here, the tumor cells, we have an antigen. When the tumor cells and antigen are ingested by the
स्लाइड
antigen,
चाहिँ देखाएको छैन स्नेहा स्लाइड सेयर गरे छैन ल अहिले आयो आयो आयो
in case of induction of T cell response to tumor, we have broadly divided into two categories. The first is induction of anti-tumor T cell response, and second one effector phase of anti-tumor T lymphocyte response. Here we have a tumor cell with tumor antigen, and the tumor cells and antigens are ingested by antigen presenting cell. And the antigen presenting cell, we have dendritic cell, B cell, macrophages. And once when it is phagocytosed, the tumor cells, it presents that tumor antigen to CD8 plus T cell with the help of co-stimulator. And besides that, this tumor antigen is also presented to CD4 plus T cell by the help of CD4TL, that is ligand. And once it binds to CD4TL, it releases the cytokines. And cytokines are the protein molecule which are secreted by both immune and non-immune cells. And its function is to, it involves in activation, differentiation, and proliferation of the cell. So it results in differentiation of that tumor-specific T cell. And once it undergoes differentiation process, this tumor-specific CD8 plus T lymphocyte cell recognize the tumor cell and will kill the tumor cells. Besides that, evasion of immune response by tumor involve intrinsic mechanism of immune evasion by tumor cells and extrinsic cellular suppression of anti-tumor immunity. So first of all, let us look at intrinsic mechanism. In intrinsic mechanism of immune evasion, the tumor may lose the expression of antigen that may elicit the immune response. The tumor antigen may be inaccessible to immune system. Tumor may fail to induce strong effective T cell response. Tumor may engage molecule that inhibit immune response, and finally, secreted product of tumor cell may suppress the anti-tumor immune response. So, in the figure is showing the mechanism by which tumor cells escapes the immune defense. The first is the intrinsic. Here in the down we have intrinsic mechanism of immune evasion by tumor, and the upper one is extrinsic mechanism of anti-tumor immunity. So in case of extrinsic mechanism, this anti-tumor immunity develops when T cell recognize the tumor antigen and gets activated and start killing the target cell. And in the immune evasion by the tumor, the failure to produce the tumor antigen due to antigen loss variant of tumor cell. Second, mutation in MHC gene that may lead to lack of T cell recognition and lack of killing the target cell. And the third one, that is production of immunosuppressive cytokines. And the immunosuppressive cytokine is leading to inhibition of the T cell activation and leading to failure of the killing of the target cell. And the extrinsic cellular suppression of anti-tumor immunity, the cell population that are involved are tumor-associated macrophages and myeloid suppressor cells that is found in most of the solid tumors that may promote the tumor growth and invasiveness by altering the tissue microenvironment and by suppressing the T cell response. And another is regulatory T cell that may suppress the T cell response to the tumors. That's why this immune system is both a cancer promoter as well as suppressor. And these are my references, and thank you. Now I'd like to welcome the another presenter, that is Sapna.
Now, further, I am going to present my topic, that is the role of growth factors and their receptors in the cancer. For the overview, here is the introduction, mechanism of the growth factor, role of growth factor in the cancer, and the references. Starting with the introduction, that is the growth factor is either polypeptide or a protein molecule made by the body, and its function is to regulate the cell division and cell survival. They are mitotic to the target cells, that is, they start the mitosis in a cell. They can initiate the cell migration, differentiation, and the tissue remodeling. May be involved in various stages of the wound healing. They can affect many different types of the cells, maybe the hematopoietic cells, epithelial cells, nervous system, or the mesenchymal tissues. They also have a responsibility of regulating differentiation of the stem cells to form the various types of the mature cells. Not only the stimulating, but some of the growth factors may be inhibited to the certain cells. Example, the TGF beta, that is transforming growth factor beta. Oncogenes code for the production of the growth factors and their receptors also. Therefore, chronic exposure of the target cells to increased amounts of a growth factor, or maybe the decreased amount of the growth inhibitory factor, may alter the balance of cellular growth. And these growth factors are crucial for the normal cellular functions, and but when they are dysregulated, can cause a cancer. They are involved in differentiation and essential to the normal cell cycle, and that is from the and thus play a vital role from the conception to the death. Here are the list of the growth factor with their functions, and some of the major are listed below. These are the epidermal growth factor that stimulates growth of many epidermal and epithelial cells. Similarly, the erythropoietin regulates development of the early erythropoietic cells. Fibroblast growth factor, that is promote proliferation of the many different cells. Interleukins, they exert a variety of the effects on the cells of the immune system. Nerve growth factor, the trophic effect of certain neurons. Similarly, the platelet derived growth factor stimulates the growth of mesenchymal and the glial cells. Transforming growth factor and transforming growth factor alpha and the beta having the stimulatory as well as the inhibitory effects on the certain cells. And the chronic exposure to either the increased amounts of these growth factors or decreased amount of a growth inhibitory factor can alter the balance of the cellular growth and may cause cancer. Let's see some of the growth factor receptors and their mechanism. And the risk factors are usually the transmembrane proteins. Growth factors receptors are usually the transmembrane proteins, with both having the extracellular as well as the cytoplasmic domains. And cytoplasmic domains have the kinase activity. Let's see towards the picture. That is, this picture has shown a signaling pathway that is related to the cell division and regulation of the cell cycle. First of all, the growth factor, that is the signaling molecule similar to that of the hormones, or a protein that binds to the cell surface receptor on the plasma membrane, initiating the pathway. And the cell surface receptor is embedded in the cell membrane. Upon binding to the growth factor, upon binding with the growth factor, that will activate the series of intracellular signaling events. The protein kinase cascade that will stimulate, and a sequence of phosphorylation events occurring inside the cytoplasm will each kinase activates into that will activates the next protein of the cascade by transforming the phosphate groups, that the phosphate group denoted by the P, and amplifying the signal. And after the amplification, the signal will be transferred towards the nuclear signal transmission will occur through the nuclear pore. And then the role of the RB and E2F proteins inside the nucleus. The RB protein, retinoblastoma protein, binds with E2F transcription factor, inhibiting in its unphosphorylated state. When the RB phosphorylated, it releases the E2F, and E2F, after it becomes active, promotes the transcription of the genes required for the cell division.
And finally, the outcome will be activation of the E2F leads to the expression of the genes necessary for the chromosome replication and other processes essential for the cell division. And misregulation of these pathways is often associated with the cancer. And this picture has shown the network of the events occurring in between the organelles. For example, organs. For example, the hypothalamus that is shown in the red, that will regulate the pituitary gland through the signals, and it will send the signals to the anterior pituitary lobe, which will release the growth hormone like somatotropin into the bloodstream, and the growth hormone and the anterior pituitary again secretes the somatotropin, which travels to the target organs such as liver or the bones. Liver, in response to the growth hormone, that will produce the growth factors, including the insulin-like growth factor. And the summary of this includes the cellular repair, regeneration, and maintaining the overall physiological functions. And this diagram also shows how the hypothalamus, pituitary gland, liver, as well as the organs work together to produce the growth factors and that regulate the bodily functions like cellular repair and regeneration. Similarly, the mechanism of action of hormones can occur in three ways, as mainly the endocrine function similar to that of the hormone action, that is, the site of action is different from the site of their production, and they are carried to the site of action to their site through the blood. And next can be the autocrine action, where their site of action and the production are the same, and the paracrine action synthesized in the certain cells and secreted from them to affect their neighboring cells. And the mechanism of action of the growth factor occurs usually after the growth factor bind to the receptors on the cell surface, with the result of activating the cellular proliferation and differentiation, as well as the promoting the growth. This leads to the activation of the different pathways, which we will see in detail by the activation. For example, the PI3K, AKT, or RAS MAPK pathways, with the activation of these signaling pathways, which we are going to see in detail. This picture has shown how external signals such as the growth factor regulate the cell cycle through the signal transduction pathways. The growth factor is a receptor protein located on the membrane of the cell, and signal transduction pathway after the binding of the growth factor activates the receptor protein, which in turn activates the cascade of intracellular proteins. And this forms a signal transduction pathway that transmits the signal inside the cell, and activation of the cell cycle control system, that the signal ultimately reaches the cell cycle and the control system, which is regulated, which is a regulatory mechanism governing the process of the cell, by which we have already seen in the previous slides also, through the checkpoints. That is, the G1 checkpoint ensures the conditions for the favorable to the cell to proceed to the next phase for the DNA synthesis or not. If the signal from the growth factor is not present or the conditions are unfavorable, that is, the cell may halt its process at its checkpoint. And this picture has shown the major signaling pathway activated by the growth factors and their receptors. That is, this image illustrates the multiple signaling triggered by the receptor tyrosine kinase and the transforming growth factor beta receptors in regulating the cellular process such as survival, apoptosis, metabolism, and gene regulation. And first of all, let's start with one of the mechanism, that is receptor tyrosine kinase. Are membrane-bound receptors activated by the extracellular signals, that is growth factors. They initiate the intracellular signaling cascade through the phosphorylation events, that is PI3K AKT pathway, responsible for the survival and mechanism. PI3K is activated by the tyrosine kinase and generates the signals, and AKT activated via PTK1 and mTOR2 promotes the cell survival and inhibits apoptosis by phosphorylating the target such as BAD, that is a pro-apoptotic protein, and negative regulators like P10 and INPP4B inhibit the pathway, ensuring the proper control. And similarly, the RAS MAPK pathway, in this pathway, that the tyrosine kinase activates the RAS, which triggers a cascade involving RAF and MEK1-2 and ERK1 and 2. ERK1 and 2 then enters the nucleus and activates the transcription factors. Example, that is FOCMYC, to regulate the genes involving in the cell proliferation and growth. And similarly, the PLC, gamma, and PKC pathway. In this pathway, there is a phospholipase. Gamma is activated downstream to the tyrosine kinase, leading to the production of the signaling molecules that activate again PKC. The PKC regulates processes like gene expression and nuclear factor beta. Nuclear factor beta is activated through the PKC or other mechanisms and translocates to the nucleus to regulate the genes involved in inflammation, survival, as well as the proliferation. Similarly, the TGF, that is the transforming growth factor beta on the right side, is signaling will be activated by the SMAD proteins. Example, SMAD2, 3, that forms complexes with SMAD4 and regulate the gene expression. And similarly, the negative regulators like SMAD6 or 7 inhibit this pathway to prevent overactivation. And when pro-apoptotic signals dominate, for example, dephosphorylated BAD, mitochondria releases the cytochrome C, triggering caspase 9 activation and apoptosis. And misregulation of these pathways is often linked with the disease like cancer. That is, overexpression or mutations of the growth factors or receptors promote tumor reogenesis. Let's see the epidermal growth factor in detail. That has a receptor epidermal growth factor receptor, as well as the other receptors are also called ERB1 and SER1. Their normal role is for promoting epithelial proliferation and wound healing. And the cancer link is overexpression and mutation in the lung, breast, and colorectal cancers. And targeted therapies can be Zeftinib and Erlotinib. These are the that is epidermal growth factor receptors inhibitors. And the right side, we can see the pathway. If we see this pathway in detail, that is the this shows the epidermal growth factor signaling pathway, which is critical for the cell proliferation, survival, and metastasis in cancer biology. In this picture, a ligand, we can see the orange color that binds to the EGFR, that is epidermal growth factor receptor, that is located on the plasma membrane of the cell. And the receptor, the green color, receptor tyrosine kinase, that is shown in the green color, that is activated upon the ligand binding, that is EGFR binding. Then this activation will lead to the receptor dimerization, pairing, and autophosphorylation that is shown in the P and red marks on the receptor, that will trigger the different intracellular signaling cascades. Receptors, for example, and in this picture also it has shown the epidermal growth factor receptor inhibitor, that is Zeftinib and Erlotinib. These are the receptors and can be used as a targeted therapies. And downstream signaling pathway, after the activation of the EGFR receptor, multiple pathways are activated inside the cell. For example, RAS, RAF, MAPK pathway. Similarly, the JAK STAT pathway and PI3K AKT pathway, responsible for promoting survival and proliferation, inhibiting apoptosis, programmed cell death, etc. And these pathway collectively promote the proliferation, cell survival, and metastasis, making the EGFR signaling pathway a key target in the cancer therapy. Similarly, next is erythropoietin having the receptor on the cell membrane, that is erythropoietin receptor, and for the normal role it has this normal role as that stimulates the cell red cell production in the bone marrow, and its receptor will be overexpression in the tumors, example breast and the ovarian cancers, and enhances angiogenesis and resistance to apoptosis. This diagram, which is crucial for promoting the survival, differentiation, and proliferation of erythroid progenitor cells, that is in red color, that is the erythropoietin or red ligand molecule EPO that binds to the EPOR that is present in the plasma membrane. And the receptor, the green color, forms a pair upon the EPO binding and activates the downstream intercellular signaling cascades. Similarly, the JAK2, Janus kinase 2, and three pathways, that is RAS, RAF, MEK, RK pathway, PI3K, AKT pathway, as well as the JAK2 STAT5 pathway. And outcome of this pathway will be the proliferation, that is activation of the ERK1 and 2 pathway leads to the increased cell division. And differentiation, the pathway collectively ensure the progenitor cells to mature into functional erythrocytes. Similarly, the interleukins receptors, maybe the interleukin 6R and interleukin 6 for interleukin 6, that is responsible for immune responses and inflammation. Interleukin 6 promotes the chronic inflammation and tumor growth, a role in cancers like the multiple myeloma and hepatocellular carcinoma. Next is nerve growth factor. Receptors are the tropomyosin related kinases, TRKS, TRKA, and P75 neurotropin receptor that will promote neuronal survival and differentiation. And this receptor activation linked with the breast cancer, breast, prostate, and pancreatic cancers enhances metastasis as well as the cancer survival. Signaling pathways activated by the nerve growth factors. This also has, after the nerve growth factor binding to its receptor, mediates proliferation, differentiation, and survival via the activation of these three pathways. And this extends to driving invasion, metastasis, and autophagic cell death in certain cancer cell types. And NGF binding to the P75 receptor initiates recruitment of the various adapters which activate nuclear factor kappa B and J and K, that is N-terminal kinase, and these mediates opposing effects of survival and apoptosis respectively. And similarly, next is the platelet-derived growth factor responsible for the mesenchymal cell growth and repair. Platelet-derived growth factor exerts its effects by binding to the specific cell surface receptors known as its PDGF receptors, which are also the receptor tyrosine kinases, and receptors are alpha and beta. Upon binding of PDGF to its receptors, downstream signaling pathways like RAS MAPK pathway, which regulates the cell proliferation differentiation, as well as the PI3K AKT pathway, which is involved in cell survival and migration, will activate. And cancer link, that is aberrant signaling, promotes angiogenesis and stromal support in glioblastoma and sarcoma. Targeted therapies may be the PG, the platelet-derived growth factor co-receptors inhibitors, that is imatinib. Transforming growth factor alpha, it has its receptor that will stimulate cell proliferation, and overexpression drives autocrine growth signaling in cancers like lung and as well as the pancreatic tumors. And transforming growth factor beta, it is one of the growth factor responsible for inhibition. It has its function as inhibition, that is members of transforming growth factor exerts effects by binding to the beta receptors, TGF beta receptors, and these receptors are serine and threonine kinases consisting of type 1 and type 2 receptors. Upon ligand binding, the type 1 receptor phosphorylates and activates the type 2 receptor phosphorylates and activates the type 1 receptor, initiating the intercellular signaling pathway, that is, and overexpression of this leads to the autocrine growth signaling in cancers like lung as well as the um, pancreatic tumors. And this picture illustrates the mechanism of the progression of the cancer from initial mutation to the mitosis and role of the different growth factors at different places. And here is a step by step explanation. First of all, let's see towards the driver mutation, that is a genetic mutation in a single cell causes it to grow uncontrollably. That is, this mutation is referred to as a driver mutation because it initiates the tumor formation. And the second part, the mutated cell begins to multiply. And the second number, that is the clonal expansion, multiply, forming a group of identical cells or clones, that is a growth factors, like in this case the insulin like growth factor 1 epidermal growth factor contribute to this expansion. Similarly, third number, that is a carcinoma in situ or intraepithelial neoplasia, that is the tumor grows but remains confined to this tissue of origin, and it is not yet invasive, and meaning it hasn't spread to the nearby tissues. And for the invasion, that is, the tumor invades surrounding tissues, breaking through the basement membrane. Molecules such as tumor growth transforming growth factor beta, hepatocyte growth factor, fibroblast growth factor, and EGF that facilitate this process. And the fifth number, dissemination, that is the tumor cells enter the blood vessels, in this case arterial capillaries or the lymphatic vessels, spreading to the other parts of the body. And in the sixth number, there is micrometastasis, that is the small cluster of the tumor cells called micrometastasis form in the distant tissues or organs. And tissues or the organs, and some tumor cells develop resistance to the therapies, chemotherapy or radiotherapy, and these resistant clones are driven by the molecules like tumor growth factor alpha and NRG, that is neurozeguline, and CSF, that is the colony stimulating factor 1. And similarly, for the angiogenesis, the tumor stimulates the growth of new blood vessels, that is angiogenesis, to supply itself with the nutrients and oxygen. And factors like VEGF, that is vascular endothelial growth factor, FGF, and SBEGF, which means heparin binding EGF like growth factor, promotes this process. And similarly, and the ninth number, the metastasis, the tumor cells colonize to the distant organs and forming secondary tumors, and this is the most advanced and often the fatal stages of the cancer progression. And this picture has shown the growth factors and development of metastasis in the major organs, and the growth factors and development of metastasis in major organs, that is the most common metastasis sites from the solid tumors are the lung, liver, brain, bone, and as well as the lymph node. And many growth factors are involved in the development of the metastatic tumors in these organs, and the organ-specific metastasis and the growth factors are listed in this picture. For example, for the lung, we have insulin-like growth factor, tumor necrosis factor, TGF beta, that is transforming growth factor beta. Similarly, the liver, we have insulin-like growth factor first, epidermal growth factor, BEGF, PDGF, tissue factor, interleukin 6, and many other. And similarly, the bone have different growth factors, brain and lymph nodes. And these are the different growth factors associated for metastasis in different major organs. And these are the references for the today's presentation. And thank you all. And thank you. And I would like to call my next presenter for today, Bisal Khatri.
Good afternoon, respected faculties. my esteemed senior, my colleagues and my juniors. So, and I also want to thanks our coordinator, ma'am, and deputy coordinator, ma'am, as well as HOD sir for this opportunities. So in today's cancer seminar, my topic is metabolic reprogramming and extracellular vesicles in cancer cells. And these are the outlines, and I will talk a little bit about all the things in respective slide. So let's start with introductions. So first of all, cancer metabolism, it has been found a foundations area of study in cancer biology, and it is emerging nearly 50 years before the identification of oncogene and tumor suppressor gene. So first of all, in metabolic sector, we have to understand about what are the metabolic reprogramming in cancer cell, because in cancer cells have to divide rapidly so that the anabolic pathway that is the anabolic pathway that runs very high in cancer cells, so that this is actually explained by Warburg. Actually, what happens in a normal cell, in presence of O2, the glucose, the metabolized glucose that runs through the glycolysis, and that will form pyruvate, and on the presence of oxygen, it will goes towards the electron transport chain in the mitochondria, and then oxidative phosphorylation. Then it produce lot of ATP in the carbon that is present in the glucose, that will convert into the carbon dioxide. So ATP will be produced. So, but when there is oxygen is limited, then cell, they will redirect the pyruvate that is generated in the glycolysis into the lactate without going to the mitochondrial oxidative phosphorylations. So Otto Warburg is a scientist, German scientist. He observed that cancer cell tend to convert most of the glucose to lactate, whether even in presence of the oxygen. So he called this phenomena as a aerobic glycolysis. We already know about the anaerobic glycolysis and aerobic glycolysis. So I don't want to talk about that. So mitochondria that remain functional in the cancer cells. And some of the oxidative phosphorylation continues in both cancer cell and the normal proliferating cells. So aerobic glycolysis is less efficient than that of the oxidative phosphorylation, because in oxidative phosphorylation, a lot of ATPs will be produced. But why the cancer cells they goes into this aerobic glycolysis, we will discuss in later slides. So this is the Warburg effect, and he won this Nobel Prize in 1931. So you can see here in this figure, there is oxidative phosphorylation, anaerobic glycolysis, as well as aerobic glycolysis. So aerobic glycolysis, that is mainly happens in the proliferating tissue as well as tumor, even in the presence of oxygen. So that pyruvate, that a lot of pyruvate which is formed in glycolysis, that will goes or shifted towards the lactate formation. This is all in this slide. So here in another figure, we can see that there are two normal cells and the cancer cells. In normal cells, we already know that glucose on glycolysis forms pyruvate, and then in TCA cycle and oxidative phosphorylation, it produces lot of ATP. And this ATP, that negative feedback, it will negatively feedback. There will be negative feedback mechanism which will stop the regulatory enzymes that in the glycolysis as well as in the TCA cycle. But in cancer cell, if we see lot of glucose, they enter into the cell through the help of the there is upregulation of the glucose transporter also. So due to which lot of glucose enters into the cell, and then this glucose goes to through the anabolic pathway, like HMP-SON pathway, different protein synthesis pathway from this phosphoglycerol also, and then fatty acid synthesis also happens in the cancer cell. So here we can see a lot of biosynthesis pathway as well as this pyruvate. Lots of pyruvate, they goes into the aerobic glycolysis to form the lactate. And here are different signaling molecules as well as different oncogenes also. They play the role to stop or to minimize the TCA cycles. So Here we can see the hypoxia inducible factors as well as protein dependent kinase as well as SNAL. It is also known as it is also a transcription factor that regulates the expression of the gene involved in the EMT. We call EMT, that is known as epithelial to mesenchymal transition, which increase the cancer cell invasion and metastasis, where there are certain cancer suppressor gene also, P53 and KIS, that is already discussed by our seniors. So I will not talk about that. And here in TCA cycle, the citrate form that will shift for the formations of the fatty acid synthesis also, and ROS that will also helps in stop the metastasis, so that also a lot of pyruvate shifted toward the lactate. So he is Dr. Otto Heinberg. He won the Nobel Prize in 1931. So this is the figure that shows the anabolic pathway. So mainly that promotes the growth. So this is the figure. We can see that a lot of glucose. This is the glycolysis pathway, and we only see the anabolic pathway because here you can see the lipid synthesis. Here you can see the nucleotide synthesis. Here you can see the one carbon metabolism, and also and also here you can see the different amino acid protein synthesis also. So that that glycolysis that runs within the cell, it will form the different from this glucose 6-phosphate, it will goes towards the SMP-SON pathway, and also this glycerylate 3-phosphate goes to the fatty acid synthesis pathway. For phosphoglycerate, that will goes to for the formation of the serine amino acid and then glycine, and it will involve in the one carbon metabolism. And here you can see the TCA cycle and then oxidative phosphorylation. It mainly shows the anabolic pathway, which is very important for the growth and proliferation in case of cancer cell. Now, what are the molecular mechanism of cancer specific metabolic reprogramming? First of all, I already discussed that the glycolysis, from glycolysis only 2 net ATP will be produced, whereas in case of the oxidative phosphorylation, over net ATP, that is 32 ATP molecules will be produced. But why would rapidly proliferating cell dependent on such unproductive pathway to produce the energy? One of the thing is that this lactate production, that is aerobic glycolysis, will be 100 times more faster than that of the oxidative phosphorylation. It is cycle and oxidative phosphorylation. So that this glycolysis, one of the reason is that glycolysis it is 100 times more faster. Another reason is that due to this lactate productions, here it is not a waste product. This lactate even can enters into the cell, and then it again reconverted back into the pyruvate, and then it will goes to the TCA cycle and produce the energy. Another reason is lactate, it when it goes outside of the cell, it creates the microenvironment of the cancer cell. It increases the pH around the cancer cell, so that it will makes the cancer cell to proliferate rapidly. So due to which the different immunosuppressor, it will also act as an immune suppression. And another reason is that in the anabolic process, we have to make different fatty acid, different lipids, different protein, and so on. So for that, we there is needed of the NADPS reducing equivalent like NADPS and NADS and so on. So for that also the glucose will convert into the lactate. So this is the signaling pathway. Here I only talks about the different molecules, different oncogenes, different proteins, protein that is already discussed by previous friends. So that here we can see different when different growth factor, they comes and binds to their respective receptor, then mainly the tyrosine kinase receptor. Then what happens? There is the signaling pathway. Ultimately that will activates the PI3 kinase pathway, which will ultimately activate the AKT kinase, that is protein kinase, and will activate upregulated the mechanistic target of rapamycin C1 protein, and it is very important protein for the growth and proliferation. And we also discuss this protein in the aging, in the nutrient signaling pathway in aging. So you can see this mTOR have different role. It also upregulated the enzymes that helps to convert the glutamine into the glutamate. so that these glutamates can enter into the TCS mitochondria, and it can convert into the alpha ketoglutarate. So it helps to run the TCA cycle also. I think this much in this slide. So I already discussed this in the previous slide, so I'm not going to discuss here. So here also in this figure also it shows the different molecules that are upregulated. So you can see there is upregulation of different transporters of this glucose and even this lactate and different amino acid also. So you can see and there are different here are different enzymes that are upregulated in glycolysis also. You can see hexokinase 1 and 2, phosphofructokinase, aldolase, phosphoglycerate mutase, as well as pyruvate kinase, lactate dehydrogenase kinase. These enzymes are upregulated so that the glucose can convert it into the pyruvate and then ultimately lactate, because lactate dehydrogenase is also upregulated. So these are the different anabolic pathway we are already discussed in the previous slide. So this is the regulation. Now, regulation of cancer metabolism. So first of all, due to this signaling, AKT, that is protein kinase B signaling, it is very important for the upregulation of the glucose transporter, different enzyme like hexokinase, and it regulates the de novo synthesis and beta oxidation. Then mTOR, this is mechanistic target of rapamycin. It is situated and helps in the signaling pathway for energy metabolism, cell growth, and proliferation. In mTOR, it is also activates the hypoxia-inducible factor 1 alpha in cancer cell. It has a different role, so we'll discuss in later slide. This hypoxia-inducible factor and its role in metabolism. So this hypoxia-inducible factor upregulates the glycolysis. Actually, it helps to increase the uptake of glucose from this transporter, glucose metabolism by increased level of glycolytic enzyme. It also helps to increase the pyruvate level, and helps in pyruvate is removed from cell by this transporter, that is monocarboxylate transporter 4. This pyruvate enters or this lactate enters into the cell by the another transporter, that is monocarboxylate transporter 1. So it also down regulates different oxidative phosphorylates लाई पनि के गर्छ, HIF ले down regulates गर्छ by decrease the pyruvate flow in TCA cycle, by decrease the mitochondria biogenesis and switch the high efficiency of cytochrome oxidase. So here this picture shows the different role of the hypoxia inducible factor. It helps to upregulate the different protein and transporter as well as enzyme in that are involved in the glycolysis or either in the TCA cycle, or help in the upregulation of the transporter of this lactate in outside of the cell. So this figure shows the role of the hypoxia inducible factor. So this is the hallmarks of cancer that links towards the metabolic changes. So here we can see this figure shows the different hallmarks of cancer, how they link to the metabolic change. Here we can see the two types of arrows. One is centripetal arrows, that is extend from center to the outside, and other is centrifugal arrows, that is extend from outside to the inside of this circle. So here in the circle there are different hallmarks of the cancer, and this outside it shows the different metabolic changes. So how they are interconnected we can see from these arrows. So how the metabolic how these hallmarks link to the metabolic changes, this can be shown by the centripetal arrows, that is extend from the center. So you can see this self-sufficiency in growth signal, ultimately it links to the metabolism by the help of the PI3 kinase, and it ultimately upregulate the AKT, that is protein kinase B, and then that mTOR activation यो चाहिँ already discuss भइसक्यो. अर्को चाहिँ यो centripetal arrows अर्को हेर्ने भने चाहिँ यो limitless replicative potential हुन्छ, किनभने क्यान्सर सेल चाहिँ एपोप्टोसिसमा जाँदैन. So because how these hallmarks change the metabolic, link to the metabolism by this, it suppress the p53 gene. It is a tumor suppressor gene, so that यसलाई डाउन रेगुलेटेड गरिसकेपछि सो अरू कुराहरू चाहिँ हुँदै जान्छ, द्याट इज ग्लाइकोलाइसिसहरू बढ्ने भयो, सो र अक्सिडेटिभ फस्फोराइलेसन चाहिँ इनिबिट हुने भयो. त्यस्तै गरेर अरू चाहिँ के छ, सबै सेन्ट्रिफ्युगल एरोज, वी क्यान सी सेन्ट्रिफ्युगल एरोज, हाउ द मेटाबोलिक चेन्जेस, हाउ दिस मेटाबोलिक चेन्जेस लिङ्क्स टु द hallmarks of the cancer. So this is already discussed by Sony. So Now, another important topic is how we measure the cancer metabolism. We can measure either by measuring the individual pathway or by multiple pathway or metabolites. So different glucose or amino acid uptake, this can be measured radioactively by determining the radioactivity of the cellular fraction. So one of the example is that glycolytic flux can be determined by using the conversion of the 5 tritium, that is hydrogen isotopes, so glucose to that will ultimately convert into the water. And by assessing the this tritium, we can determine the glycolytic flux. Another one is mitochondrial potential can be quantified by spectrophotometry or flow cytometry by using the another fluorescent dye, that is TMRE, also known as tetramethyl rhodamine ethyl ester. And ATP can also directly measure by the luminometry by using the different enzyme like luciferase or protein like luciferin. In oxygen consumption, we already know it can be measured by different oxygen electrodes, by the method called electrodes called Clark electrodes. And lactate production can be measured by using the pH change by the help of pH meter in culture media. And different metabolites can be assessed by high advanced technology like mass spectroscopy and nuclear magnetic resonance. Now, this is the last slide, last topic of today's presentation, that is extracellular vesicles. So it is also important because it is a small heterogeneous secreted vesicle, and it is important in cell-to-cell communication and helps to contribute important in both normal and pathological physiology. It is actually a lipid bilayer, which this extracellular vesicle, it is composed of lots of different molecules like lipids, DNA, RNA, small micro RNA, non-coding RNA, as well as small protein and bioactive small molecules. So these exosomes, how it is related with cancer, these exosomes can be released by the cancer cell also. So that will promote the cancer growth by delivering the signaling molecules like growth factor and oncogenic protein to the other cell. So they can stimulate the angiogenesis, or they can play the crucial role in helping cancer cell to spread to the distinct organs, and also they serve as a biomarkers also in near future for cancer diagnosis, prognosis, and so on. So this is the figure how these exosomes are formed. They are mainly formed from the microvascular body, from exocytosis, and so we also call it exosome. And another types of the exosomes we call is microvesicles. It can be formed by the membrane body, and these exosomes contain different types of molecules. I already discussed. They can be either lipid, they can be either mRNA or protein or antigen and so on, DNA and so on. So they now goes to the target organs and they bind to the receptor and they do the different cell function. So these are the highly interested molecules nowadays for research in cancer biology. So this is the summary of today's presentation, and this is the references, and thank you so much. And then I would like to call the next presenter, Dr. Pius. Thank you.
Well, good afternoon, respective faculty members, my senior, my colleague, and my junior. I'll be discussing on the role of oxidative stress in the carcinogen. So these are the objectives of my today's topic. Talking about the oxidative stress, it is a free radical. Talking about the free radical, that usually refers to the chemical species with an unpaired electron in its outer orbital, so which is highly reactive, and that usually donate or accept the unpaired electron to attain the stability. And oxidative stress is defined as the relative excess cellular oxidant molecules when compared with the antioxidants level, and has been linked to the carcinogenesis. The cellular oxidant species comprise of the usually ROS, that is the reactive oxygen species, and the RNS, that is the reactive nitrogen species, with the various oxygen containing free radicals and other unstable, highly reactive chemical entities possessing the oxygen constitute, which is termed as reactive oxygen species, such as the superoxide radical, hydroxyl radical, hydrogen peroxide, and the hypochlorite ion. While The RNS are the peroxynitrite, nitrous acid, and the nitric acid. So there are the various sources of the oxygen species in the cancer cell. The major ROS that includes the free radicals and the non-radicals, that is the hydroxyl radicals. That the source is the HCl, and whereas the superoxide radical, that the sources are the electron transport systems, and one electron reduction of the oxygen by the respiratory burst via the action of the membrane-bound NADPH oxidase, and the peroxyl radicals that is produced from the Fenton reaction, where the non-radicals are the hydrogen peroxide, hypochlorous acid, and the lipid peroxide, whereas the major RNS contains the free radicals and the non-radicals. The free radicals is the nitric oxide, and the source is from the nitric oxide synthesis, and another is the nitric dioxide, that is the source is the activated neutrophils, and the non-radicals contain the dinitrogen trioxide, oxynitrate, nitrite, and the nitrylines. These are the various sources. You can go through it. And so the basic mechanism of the oxidative stress related to the carcinogenesis are that it can be either the oxidative stress can increase either due to the environmental factor or the intercellular ROS. The environmental factor includes the ionizing radiation, UV radiation, xenobiotics, smoking, and the pollution, whereas the intercellular reactive oxidative stress can be the mitochondrial electron transport chain, cytochrome P50, NADPH oxidase, lipoxygenase, and peroxymal oxidase. The endogenous, whereas the various antioxidants can be either the endogenous or the exogenous. The endogenous can be further divided into the non-enzymatic, and the enzymatic. The non-enzymatic contains the uric acid, bilirubin, albumin, and glutathione, whereas the enzymatic contains the glutathione peroxidase, SOD, catalase, and the exogenous sources can be the vitamin C, E, or the beta carotene. So increase in the oxidative stress either can cause the DNA damage by the mutation, or the protein damage by the unfolded protein response, or the lipid peroxidation by
पियुष आवाज सुनिएन फेरि एकचोटि अनम्युट गरौँ
the oxidative stress can be either whenever there is increase in the oxidative stress, that can lead to the DNA damage, leading to the mutations in the various genes, and or it can either cause the protein damage or the lipid peroxidation. The protein damage can be caused by the unfolded protein response, and the lipid peroxidation can be caused by the membrane and the organelle damage, or the generation of the oxidative LDL, and that ultimately leads to the cancer. So there are the various transcription factors that are activated by the ROS, that are the usually activated protein 1, AP1, or HIF, hypoxic induced factor 1 alpha, heat stroke factor 1, nuclear factor kappa B, whereas the nuclear factor erythroid 2, P45 related factor 2, and the tumor protein P53 or TP53 or the TRP 53 in the mice. Proc-head box class O, that is the FOXO. Also, there is another factor, that is the peroxidome proliferator activated receptor gamma coactivator 1 alpha. So this is the ROS associated signaling pathway in the cancer biology. That the ROS mediated signaling components can be either the from the TNFR or the CD95 or the trial. There has been a number of developments and the reports published how ROS modulate the expression and the functioning of the various signaling components and the pathway associated with the cell growth and the hemostasis. We'll be much more learning about in this upcoming slide. So these are the ROS has been linked to the play multiple roles in the hallmark of the cancer. There are almost 10 hallmark of the cancer. First one is the self-sufficiency in the proliferation signals that causes the ligand in independent RTK transactivation, and there is the decrease in the RTK activation threshold, whereas the insensitivity to the anti-proliferation signals causes the activation of the P53 enzyme, gene, loss of the contact inhibition and the loss of the anchorage dependence, whereas the invasion and the metastasis usually deals with, causes the metastasis overexpression, metastasis secretion, invatophobia formation, plasticity in the motility, and the limitless replication causes the expression of the telomerase, where the continuous angiogenesis is the formation of the new vessel and causes the endothelial progenitor activator signaling the BEGF and the angiopoietin, recruitment of the perivascular cells, and also there is the another hallmark, that is the escaping apoptosis that causes the inactivation of the P10 gene, activation of the SRC gene, enoicase resistance, and the there is the change of the cell metabolism that causes the Warburg effect, upregulation of the glucose transporter, activation of the oncogene and RAS and MIC, and the mutation of the tumor suppressor gene, that is the P53, and there is increase in the HIF1. There is another hallmark is the escaping immune destruction that causes the cancer cell disable the components to the immune system and are dispatched to to eliminate them. Another hallmark is the genome instability and the mutation that causes the increase in the rates of the mutation, increase the sensitivity of the mutagenic gene agents, and compromising the surveillance system. And the last one is the tumor promoting inflammation that causes the acquisition of the mutation by the neoplastic cell. So ROS plays the multiple roles in the hallmark of the cancer. First one is the self-sufficiency in the proliferative stage, in which most of the normal cell waits for the external masses before dividing, and conversely, cancer cells often counterfeit their individual proliferative masses, and the ROS play a ligand independent RTK transactivation, decreased RTK activation threshold, insensitivity. Second one is the insensitivity anti-proliferation signals. As the tumor enlarges, it generally squeeze the tissue nearby it, and therefore receive the message that would normally stop the cell division. And the malignant cells ignore this command, and the ROS are involved in the P53 activation, leading that causes the loss of contact inhibition and the loss of anchorage dependence. And increased ROS levels contribute to the sustained level survival and proliferation through many pathways, including PI3 kinase, ATK kinase, MEPK, ERK kinase 1, 2, and the PKD. Whereas the third one is the invasion and the metastasis. Metastasis are usually in which the tumor has been outgrown from the primary area to the second other area, a part of the primary area. The cancers are usually lead to the death only after they overcome their confines to the particular organ in which they arose, and needs to escape the primary tumor so that it can invade the matrix of the other organ and find the suitable metastatic and grow in the secondary sites. and the ROS play a role in the metastasis overexpression, matrix metalloproteinase secretion, and the plasticity in the motility, where the limitless replication. We usually, the normal cells cannot divide more than 70 times, but the malignant cells can divide more than that of the 70 cycles to make a tumor. Hence, tumor needs to hence the tumor needs to enforce the reproductive limits of the cell, and the ROS involved in the expression of is usually due to the telomerase. So another is the continuous angiogenesis. The tumor is characterized by the chronic activated angiogenesis due to the unbalance of the mixed angiogenic signals, thus signaling the cancer fate. ROS play a role in the increased ROS level, and this contribute to sustained cell proliferation and through many pathways, including the PIK and various gene, and thus the ROS also inactivate the downstream target, including the BAX, BEEP, and FOXO and P10 gene and JNK pathway, and this endothelial progenitor activation signaling of the VGF and the angiopoietin recruitment of the perivascular release the VEGF, that is and ultimately causes the angiogenesis. Where the next one is the escaping apoptosis in the healthy cells. General condition including genetic damage or the lack of extracellular matrix addition activate the suicide program, that is the apoptosis. But tumor cell bypass this mechanism, thereby surviving to the death message. The toxic ROS levels damage the mitochondrial membrane, releasing the cytochrome C to the cytoplasm, which forms the complex of the APF1 and the caspase 9 gene. That usually this induced the cleavage of the caspase 3 and the 7, resulting in the apoptosis, and additionally binding to the tissue necrotic factor alpha ligand to the TNFR death receptor triggers to the activation of the caspase 8, leading to the cleavage to form the caspase 3, and also at last the caspase 8 activation also cleaves the BCL1 protein by bit to form telomerase bit, which further leads to the release of the cytochrome C in the intrinsic apoptotic pathway. The next is the change of the cell metabolism. Tumor have the capability to modify or reprogram the cellular metabolism to successfully carry on the neoplastic progression, and the ROS here involved in the Warburg effect, upregulation of the glucose transporter, activation of the oncogene, and the mutation of the tumor suppressor, that is the P53, and the increase in the hypoxia-induced factor 1. The next one is the escaping immune destruction. The tumors acquired the capability to invade natural immunological destruction by either the T and B lymphocyte, macrophage, and the natural killer cell, which there are two additional characteristics facilitating the acquisition of the aggressive feature called the enabling characteristics. Next one, the genome instability and the mutation. The genomic alteration due to the epigenetic mechanism, the increased rate of the mutation or the enhanced sensitivity to mutagenic agents can drive the tumor progression. And here there is the increased rate of the mutation, increasing the sensitivity to mutagenic gene, mutagenic agents and the compromising the surveillance system. And the last one is the tumor promoting inflammation. There is the innate immune cells which are designed to fight the infection, as you know, and they heal the wounds, are inadequately support the acquisition of the hallmark capabilities, which leads to the tumor expansion. And ROS are involved in the acquisition of the mutation by the neoplastic, thus accelerating their evolution towards the heightened malignancy. So there is also the role of the ROS in autophagy. The autophagy is usually the consumption of body on tissue during the metabolism. What happens? And the autophagy generally happens in all three stages. That is the initiation, promotion, progression, and the metastasis. And this during the initiation, the autophagy is the anti-tumoral, but after the initiation, in the promotion, progression, and the metastasis, then it acts as the pro-tumoral. The ROS is in the cancer depends on the tissue and the stage on the type of the tumor. During the early stage of, as I have already told, in the autophagy plays a protective role in the preventing ROS-induced DNA damage on DNA and protein, leading to the inhibition of the tumorigenesis. During the late state, again, that is the promotion, progression, and the metastasis, autophagy presents at the pro-tumoral role, and through the elimination of the ROS-induced metabolic stress and the production of the nutrients required for the cancer survival. During the development of the tumor, primary tumor, a strong interaction between the stroma cell of the microenvironment and the cancer has been observed, and these cancer cells under hypoxia induce the formation of ROS, which can activate the autophagy in the stromal cells. These cells when then provide high energetic nutrients such as the lactate, ketones, and the necessary of the cancer cell survival and the proliferation. So this is the various stage of the cancer, that is the initiation, promotion, and the metastasis. During the initiation stage, the disease stage is not the acquisition of the genetic change sufficient for the autonomous cell growth, and there is increased positive oxygen O2, H2O2, hydrogen peroxide. And in the promotion stage, there is a proliferation of the pre-neoplastic nodules that have acquired autonomous growth, and there is increase in the hydrogen peroxide employed as the signaling molecule that mediates cell proliferation. And in the progression stage, as the cancer progresses, there is the local invasion of the tumor into the adjacent normal tissue with anchorage dependent and the interaction with the stromal cell, also the immune cell. And there is the effect of the oxidative stress on the different stage of the cancer, that there is the increase and decrease, either positive or the negative, upregulated or downregulated of the H2O hydrogen peroxide, likely supports the EMT and the tumorigenic action of the tumor associated macrophage, but O2 and used by the immune cells to kill tumor cells. And lastly, in the metastatic disease, the dispersal of the tumor cells via blood stream into other colonized distant organ. There's a negative high that is down regulated, and there is a high oxygen level in the circulation trigger apoptosis in certain types of cell of the tumors. So lastly, the ROS and the radiation therapy. This is just whenever we take the radiation therapy in the cancer, sometimes it might induce the for the treatment of the, certain cancer, it may cause the another cancer, leading to the another cancer. That is, the radiation therapy used in the treatment of certain malignancy renders its effect on the cancer cells due to the toxicity generated by the free radicals. The radiation used in the radiation therapy leads to the generation of the oxygen-derived free radicals in the tumor environment by ionizing intracellular molecules like oxygen and the water, and thus result in the death of the cell, mostly due to the extensive membrane and the DNA damage. So these are the difference. Thank you. I'd like to call next presenter, Dr. Roshan Thapa. Thank you.
Good afternoon, respected faculty. Good afternoon, respected faculty, my senior, my colleague, and my senior. Today I'm going to talk on the topic tumor markers. These are my overview. Tumor markers, introduction, target tumor markers, role of tumor markers, current clinical applications of tumor markers.
Dr. Roshan, can you make it a little loud?
Okay, ma'am.
Speak little loud.
So these are my overviews, the tumor markers. In introduction, the ideal tumor marker, role of tumor markers, and present or current clinical application of the tumor marker, and classification of the tumor marker. Now we're going to talk about the introduction part, the tumor marker. These are the substances that are present in and produced by the tumor or by the host in response to the tumor. They are usually present in abnormal high concentration in the body fluid or tissue from the patient with cancer. They are usually found in cells, tissue, or body fluid, are measured qualitatively and quantitatively by chemical, immunologic, genomic, and protein methods. They are also used to identify the likely presence of a cancer and also to differentiate between the tumor from the normal tissue. What is known as the ideal tumor marker? They are usually detectable only in the given the malignancy or absence in the healthy population, or not present in the non-malignant population. Usually it is done with the help of the clinical specificity and clinical sensitivity, which approach about the 100%. They are useful to consider the requirement of the ideal tumor marker. The following are detectable only in presence of malignancy, which are present in, and they are also present in readily acquired biological metrics such as serum, urine, tumor marker, CSF, so on. And they are also present at the concentration that are relatively high proportion to the tumor burden. They are conveniently measured by the readily available, simple, reproducible, and inexpensive procedure also. And they are usually built to the clinical cure with measurable effect on the patient outcome. These are the ideal tumor markers. Now we will talk about the roles of the tumor marker. What are the roles of the tumor marker? They are usually for the early detection, usually for the diagnosis confirmation, and also in order to find out the stage and prognosis of the tumor, and also for therapy monitoring and recurrence detection. Some tumor markers, they can provide only warning signs for the specific cancer. For example, the person having the bone cancer or bone tumor, usually present with back pain or bone pain like that, and usually you can find from the clinical diagnosis also. And usually, to diagnose the, to confirm the diagnosis, usually do the certain diagnostic tools like imaging, biopsies, PET scan, MRI, like so on, and CT scan also. And in order to find the staging and prognosis, which helps to determine the extent of the cancer and likely cause. And they're also useful for the therapy monitoring, which measures the effectiveness of the ongoing therapy, ongoing treatment. And also, to find out that there is recurrence or not, to identify the cancer recurrence or the clinical treatment. Now we're going to talk about the current clinical application of the tumor marker in treatment area. They are screening for the cancer, for diagnosis of cancer, for assessing the prognosis, prediction of the treatment response, and monitoring the treatment. And also they are used for monitoring the disease, post-treatment to detect the recurrence. Here you can see in the picture. They utility is screening, diagnosis, prognosis, monitoring treatment, and recurrence also. First, any utility that biopsy indication, then high level indicates that there is a presence of disease. And again, if there is a high level activity for prognosis, then researchers that are used for the indication of the chemotherapy. We can see them below there. And also for the monitoring the treatment, to monitor efficacy of the chemotherapy and this will be used after the surgery. And detection of the recurrence, which is by you can find out with the increase, it will use the relaxer. And also about the tumor marker, you can see the time how it is screening diagnostic and so on. You can see the PSA and with the time with the netonafrin, LPA, like that, and chromogan A to the full, yes, and like so on. For the recurrence and pollution also we use to find the tumor marker, like we take to the help of the tumor marker. Now can be the cancer specific or tissue specific. For the cancer specific, specific of the certain cancer tissue got large overlap, which means low specificity. And for the tissue specific, we use the PSA, of known as the specific antigen, which is produced by the prostate, and also from the alpha-fetal protein, which is usually produced by the liver, and beta F is known as the beta chain of the chronic gonadotrophic hormone. So beta F is the hormone. Beta chain of the chronic gonadotropin, which is produced by the embryo, and like so on, thyroglobulin, which is produced by the thyroid. Now we will talk about the classification of the tumor molecule. Then you can see where the hormones, enzymes, and protein and polypeptides and tumor antigen take on the classification tumor. So on there are other classifications based on the nucleotide, IgG or the hemoglobin, receptor, oncogene product, and various markers. And here we can talk about the enzymes as a tumor marker. What usually it does is almost the tumor, even almost the serum enzyme. In the main critical either with the absorption of increased amount of enzyme by tumor cells or due to the release of intercellular enzymes as a result of the cell damage. Now we're going to see in the picture here, the tumor markers, tumor size, nature, specimen, and clinical utility. First we go from the PSA, something known as the prostate antigen, prostate specific antigen. Usually the tumor type here is prostate cancer, usually drawn by the illness in the lab. And what we need to mean is to serum here, all the PSA, LDS, ALP, and all we need to serum for the tumor marker diagnosis or for the detection. And PSA for the prostate cancer, whereas LDS for the hematologic malignancy, and usually drawn by the method known as the enzyme assay. And what it does is prognostic indicator, it shows the elevated non-specific especially in the numerous cancer, and ALP also known as alkaline phosphate, which shows the tumor type of metastatic cancer, metastatic carcinoma, bone, SCC, cellular carcinoma, sarcoma, lymphoma, leukemia. And the method used here is enzyme assay, when we use the serum for the specimen. Usually it helps to detection of the liver and bone involvement, and also the many bone-related and liver cancer. And it's also known as the neuron-specific stimuli, which is helped to detect the neuroendocrine tumor. It can be detected by the RIA, also known as radioimmunoassay or IFC immunohistochemistry, which shows the progression of the neuroendocrine tumor. Now we're going to talk about the hormone as a tumor marker. Here there are so many tumor markers like the CTR, KDR, etc. calcium, chromogan A. Hello, sorry, there is a problem with me too. Now I'm going to continue with the hormones as a tumor marker. Here the CTR is also known as adrenocorticotropic hormone, which leads to identify the pituitary adenoma, specific
Just
Dr.
Roshan, just list the tumor marker and tumor type of that hormone, okay? Don't tell about method, serum, clinical utility, and all those things. Just tell these two things.
Okay. Okay, one thing. So this is the tumor marker, which are used to identify the protein the tumor type. They are like adenocorticotropic hormone, which is used to identify the pituitary adenoma, SPS producing tumor. And so there is the antidiuretic hormone, which is used to identify the posterior pituitary hormone, like peptide hormone, which is used to identify the insulin secreting hormone. And calcitonin, which is for the medullary thyroid, medullary thyroid hormone, and neuroendocrine also. And likewise, chromogranin A, the tumor marker, which is used to identify the pheochromocytoma, neuroblastoma, plasma tumor, and mostly lung cancer. Likewise, the cortisol, which is used to identify the adrenal tumor, and likewise, gastrin hormone is also used for the diagnosis of neuroendocrine tumor. And growth hormone is used to identify the pituitary adenoma, growth hormone secreting tumor. These all are identified by the serum. Now we're going to talk about two or three tumor markers which can be drawn by the specimen of the urine, either of the 24-hour urine and from the plasma muscle, like CME, which is also known as phenylneuronuric acid, and here also known as prolactin. Now I'm going to continue here. NSB, balancing acid, also used to diagnose the neuroblastoma, osteochromocytoma, and paraganglioma. Down in the list, it has also called in specimen. Likewise, 5-SIAA, also known as the 5-hydroxyindole acid, which is used to diagnose the plasma tumor. And likewise, there is also the PCS, which is used to identify the thyroid hormone, thyroid and prolactin, which to identify the pituitary adenoma, and DNA also known as phenylneuronuric acid, phenylneuronuric acid, which to identify the pheochromocytoma, paraganglioma, and neuroblastoma. Usually, is used as well. Lipid chromatography, SPG and 5-SIA answers, IA and methylation as well. Now we're going to talk about the antigen at the tumor marker. Here there are different tumor markers like CA199, 163, and 23.1, and CA125. They are usually the antigen, cancer antigen, which is to diagnose or to identify the certain diagnosis tumor marker. For the CA199, which is used to monitor the cell cancer, and usually tumor type is gastrointestinal cancer and urinary carcinoma. Likewise, the cancer antigen antibody, which is the type of metastatic breast cancer phenotype, and what you see find out the recurrence and the specific class. Likewise, the cancer antigen, which has the phenotype of metastatic breast cancer, and cancer antigen on the 25, which is the phenotype of ovarian cancer. And they are also used for monitoring the therapy. Now I'm going to talk about the receptor epithelial marker. With the help of these things, you can find out the tumor marker, like there is the epithelial receptor, projection receptor, or HER2, or any when or specifically the growth factor receptors. These receptors, they are usually drawn by the biopsy method, and usually the method used here is immunohistochemistry, and these are unknown therapy or receptors. They are used to find out the hormone therapy indicator also. Now we're going to talk about the estrogen sector and the position sector. You see, there is a phenotype of breast cancer and HCR2 and NEU, like NEUs, which have phenotype of breast, ovarian, and gastrointestinal tumor. Likewise, there's the epidermal factor receptors, which has a phenotype of head, neck, ovarian, and cervical cancer. Now we'll talk about the serum protein and peptide and the clone material. Here, some marker you can see the serum and protein, serum say lysine and beta-2 methylchlorine. Usually, the serum and protein is the clone type of the plasma serum of patches, patches, and these all are done by the treatment of the serum. And for the serum and protein, it's used to the initiation and routine monitoring of the plasma serum and everything. Likewise, there is a serum-free light chain, which also has a more type of the plasma serum. This process, and we get a tumor type of methodology and here and it is the clinical components of the kind of the chronic marker of the which is called lipopolysaccharic disorder. These are my friends. Thank you. Now I'd like to call my next presenter, Dr.
Okay, am I audible right now?
Yes, yes.
Okay, so we are at this stage of treatment here. Cancer has been diagnosed, patient has gone to the doctor for the treatment. Now, what are the different treatment modalities that he is going to receive? We'll be seeing with this presentation. So here, for this treatment of cancer, there are many types. But some people, they receive only one type of treatment, whereas some people, they might receive combination of treatments. And the treatment type, it depends
Dr.
on
Dilip.
the type of the cancer.
Dr.
Yes,
Dilip. Excuse
yes,
me.
ma'am.
There is something coming in your slide. Other people couldn't saw is the slide ma केही देखिरहेको हो त्यो कभर गरिरहेछ कि केसरी अब भयो okay now it's okay.
So, coming to the treatment types, there are many types. Some patient may receive one type of treatment, whereas some cancer patient, they might receive combination of treatment. And the treatment type is determined by the type of the cancer that the patient has been diagnosed. So coming to this treatment type, there are, let's see, the different modalities of treatment. For example, here is biomarker testing can be done. Second, chemotherapy. Here we use the drugs to kill the cancer cells. Then third is the hormone therapy. Then hypothermia. So here the body temperature is raised in cancer patient to treat them, and then immunotherapy, photodynamic therapy, radiation therapy, stem transplant therapy, surgery therapy, and targeted therapy. So these are the overall therapies for the cancer patient. Here in this seminar, I'll mainly focus on the immunotherapy and the chemotherapy. So here, let's see this chart here. The cancer, after the diagnosis, we have to find out whether the cancer has been localized in one location or it has been distributed throughout the body. If the cancer is localized at one organ, then the treatment modality can be surgery or radiotherapy. If the cancer has been metastasis to different distant organ, then the doctor suggests of this systemic therapy to cure the cancer. And under the systemic therapy, the following therapies, it comes, for example, cytotoxic therapy or chemotherapy, hormone therapy, targeted therapy, and immunotherapy. So these are the various treatment modalities that comes under the systemic therapy when the cancer has been metastasis to different organs in a patient. So coming to this chemotherapy, it means we are killing cancer cell with the help of some drugs. That's why it is called chemotherapy or chemical therapy, and in short, it is also called chemo. So basically, doctor advised this chemotherapy for two reasons. One is to treat the cancer, and other is to relieve cancer symptoms. So the doctor, they decide which drugs to use to the patient. So it depends upon the type of cancer, whether or not you had the chemotherapy before, or is there any health problem to the cancer patient, such as diabetes or heart disease. So these are some factors that doctor might consider before giving the chemotherapy. So here are some terms that we often come under, like when a patient receives chemotherapy. For example, curative, neoadjuvant, and adjuvant. So curative, by its term, it means total cure. So after receiving this kind of drugs, the patient is cancer-free. Whereas in neoadjuvant, it means, just see here in the diagram, this cancer cell is too big to operate. So what do the doctor do? First, they prescribe this chemotherapy. Why? To decrease the size of this cancer. So you can see here the cancer size has been decreased. And after that, the doctor do the surgery. So hence the chemicals that has been used in this process, it is called neoadjuvant chemotherapy, and the other is the adjuvant therapy. So here it means to help, like after some patient, they have undergone the treatment like some surgery, then the patient might receive this chemotherapy to cure. And this adjuvant chemotherapy is also done to eliminate this micrometastasis. My previous speaker, they have already explained about this micrometastasis, so I will not go much more about it. So this adjuvant therapy is to eliminate micrometastasis, and adjuvant therapy is also means to, in addition to or after the main treatment. And there is one more word, combination therapy. So most of the patient, they might receive chemicals, chemotherapy plus some other therapies. So here in the example, you can see patient is receiving this chemical therapy as well as this radiation therapy. So this kind of therapy is called combination chemotherapy. And other one is the palliative therapy. It means the cancer has already spread to the almost all the organs of the body at the time of diagnosis. So it is very impossible for the doctor to get the localized treatments of such as this surgery or radiotherapy. So in that cases, what the doctor they suggest? Palliative chemotherapy, just to relieve the symptoms of symptoms of the patient and to extend its life. So I have already told about this slide how chemotherapy has been used, such as it has been used in called neoadjuvant, adjuvant, and it also works with other treatment. So here in this slide, we can see what are the various ways we can give drugs to the patient. For example, we can give drugs via oral or via IV or some other ways, such as intramuscular, subcutaneous, intrathecal, intracavitary, topical. So basically, most popular modality is the intravenous. So these are the various different methods by which the doctor prescribe this chemotherapy to the patient. So drugs might be given through IV cannula. If someone is afraid of IV cannula or need to receive this treatment for longer time, like 1 year, 2 year, 3 year, the doctor might suggest this PICC line, which you can see in the diagram, this line. So here the catheter is directly through this brachial vein. It reaches to the right atrium. So this is called the PICC line, or Hickman line is also there, or no, Potacat. So these all modalities has been used if a patient is receiving chemotherapy for longer period of time, so that they don't need to prick very often. So now let's see the principle behind this chemotherapy. So how does this work? So here you can see the diagram. Chemotherapy means cytotoxic drugs is there via infusion. It is given to the blood vessels, and through blood it is distributed throughout the body, and it will reach to the cancer cell and it will do its job, means it will kill the cancer cell. So how it does? So it does by this rapidly, so it target this rapidly dividing cell. So whenever these chemicals reach to the particular cancer cell, it will target this rapidly dividing cells. Hence, it will helps in the cancer treatment. So this leads to cell death and shrinkage of the tumors. The tumor size gets reduced. So here you can see the list of these cytotoxic drugs which has been prescribed to the cancer patient for its treatment. So it will be very difficult for us to remember all. So let's see this drugs, its principle in this diagram here. So this is the normal cell cycle, which we have already, my previous speaker, they have already explained us. So this cell cycle, this is the normal. In every individual, this cell cycle is going on. So it is almost taking one cell cycle. It takes almost 24 hours. So G1 phase, S phase, G2 phase, or M phase. So what do this cancer means? Uncontrolled growth. Means this cycle is running very fast. So we have to check the growth. So it means we have to hinder this particular any cycle here. So here you can see interphase is divided into three phases, G1, S, and G2, and this is the M phase. So there are some drugs here. You can see for this taxane and vinca alkaloids, it targets in the M phase. Means in M phase there is formation of microtubules, so it will target the microtubule. Hence the microtubule will not form effectively, and the separation means that cytokinesis is hampered here. So similarly here at the phase here you can see in S phase anti-metabolites. So in S phase, you can see anti-metabolized drugs are used. It means like these are the drugs that affects the metabolism. Like anti-metabolism means synthetic phase, we need some nucleotides to synthesize like a new strand of DNA. So this kind of drugs, they will particularly affect at this synthesis process of this DNA synthesis phase. And here are some drugs such as bleomycin, topoisomerase inhibitors. They affect on the G2 phase. So the principle behind is just uncontrolled जुन growth भइरहेको हुन्छ त्यसलाई चाहिँ ड्रग्सहरूले चाहिँ रोक्नु पर्यो. So here you can see the same here 6 small captopril, these drugs, it's a class of purine analog, and it's basically mechanism is it will lead to the formation of IMP to the AMP. Similarly, there are drugs here you can see this vintress, which has already been discussed. It will inhibit the cytoskeleton assembly and spindle movement. So the treatment part is basically we have to focus on the particular phase. Means each cytotoxic drugs act on the particular phase of the cell cycle. So here in this slide, what you can see, this is this focus here. This is the receptor here. This is the ligand. It binds and it transfers the signal. This signal is necessary for the growth of the cancer cell. So this signal is very much necessary for the growth of the cancer cell. So here you can see after this ligand bind to the receptor, it will generate intracellular signal, and the cascade will generate down, and it will help in gene expression, cell proliferation. So what do the scientists they have discovered? Some drugs that particularly affect this signaling cascade, so that the signal is hampered. So whenever the signal is hampered, gene expression is hampered. So gene expression is hampered means there is cell growth is also hampered. So here you has been various drugs such as this OSU 031, it will inhibit this PTK1, and there's one drug sorafenib, it will inhibit this RAF, and this drug AZD624, it will inhibit this MEK. So here the basic principle is we have this drugs has been designed to target to inhibit this signaling cascade, so that this normal process of this growth of cancer cell is hampered, and hence the cancer cell cannot grow. So coming to this side effects of chemotherapy, means we have been using drugs to kill the cancer cells, but the drugs, they cannot separate which one is the cancer cell, which one is the normal cell. So what do these cytotoxic drugs do? They kill the cancer cell as well as they also kills the normal cells. So hence, what type of normal cells means? Those cells which are having high rates or high cell turnover. For example, that cell lining the mouth, intestine, or hair follicles. So basically it will affect these cells, and entirely almost all the organs will be affected. So these are the various common side effects that a person might have after this receiving cytotoxic chemotherapy. And there is one more word. This is scheduling chemotherapy. Means, okay, patient has been diagnosed with the cancer, so why we can't give like one drug, like cytotoxic drug at a time to kill the cancer cell? It because we will have a lot of adverse effect, hence the person might die because of this adverse effects. So what do the scientist or this doctor do? They will treat the patient in various cycle. एउटा डोज दिएको हुन्छ, त्यो डोजले के गरेको छ, यहाँनिर हाम्रो क्यान्सर सेल्सलाई चाहिँ नम्बर अफ सेल्सलाई कम गरेको छ, त्यसपछि रुक्ने केही समयसम्म। किन भन्दाखेरि हामीले त्यो साइड इफेक्टबाट चाहिँ हाम्रो पेसेन्टलाई चाहिँ रिलिभ गराउनु पर्छ। सो साइड इफेक्टबाट पनि रिलिभ हुन्छ, अनि साइड इफेक्ट सँगसँगै क्यान्सर सेल पनि ग्रो हुन्छ, बट द ग्रोथ हियर इज not so much. So here you can compare these two peaks. Your initial यति थियो भनेपछि now next time हाम्रो cancer cells यति छ, then coming to the next cycle फेरि cancer cells यहाँ पुग्यो, फेरि next cycle. So ultimate our goal is to reduce the number of cancer cells to 0. So this is called log kill or fraction kill. It means like with subsequent doses will reduce the constant fraction of the cancer cell, and this is called as log kill or fraction kill. So this is done to achieve via this giving chemotherapy in consecutive cycles. So, okay, the patient has received chemotherapy. Now, there are some cases that the patient might develop this drug resistance. Means that drug is not effective for them. Like how it happens? It means like So whenever any cancer cell has received this cytotoxic drugs, at that time, they contain billion of cancer cells. So significant chance is that some of those cells, there are cancer cells. So in receiving treatment, some cancer cells they might develop mutation. So this mutation help them to make insensitive to drugs. So कुनै अब 1000वटा क्यान्सर सेल छ भनेपछि ल त्यसमाबाट 10-15वटा सेल चाहिँ त्यो पार्टिकुलर ड्रगको लागि चाहिँ रेसिस्टेन्ट भइदिन्छ, मतलब रेसिस्टेन्ट भएर बस्दिन्छ. So however these resistant cells live on and go on to form tumors. So chemotherapy सक्यो but resistant cells is still there but in lower number. So त्यसपछि यो के गर्छ बिस्तारै फेरि with time it grows. So live on and go on to form tumors and that are resistant to the drug. So अब यसले जुन tumor बनाउँछ, so this tumor is resistant to the cytotoxic drug that the patient has already received. So these tumors are resistant to other drugs which have been given, and this is also known as like multi-drug resistance. This is one mechanism. Other mechanism occurs usually as result of tumors gaining mutation, means tumor constantly grow bhayirahuncha chahi there is also chances of mutation. Kasto mutation le गर्दाखेरि चाहिँ it leads to the expression of the efflux pump. मतलब भनेपछि हाम्रो जुन सेल मेम्ब्रेन मा छ एउटा efflux pump हरु चाहिँ maximum number मा आइदिन्छ अनि drugs entry गर्ने बित्तिकै efflux pump बाट drugs फेरि out भइदिन्छ. So drugs it cannot stay inside the cell, hence the drug is not effective on those cancer cells. For that reason, the doctors they use this combination therapy. So here you can see 3 vials, means 3 different drugs has been prescribed to a cancer patient, not a single drug. You know, यो 3टा drug prescribed गरेको छ भने 3टा drug को आफ्नो different mechanism छ. We have already seen in the cell cycle हामीले G1 हेर्यो, G2 phase हेर्यो, S phase हेर्यो, M phase हेर्यो. So यो जुन different drugs side of this chemotherapy drugs छ, एउटाले G phase मा act गर्छ, अर्कोले M phase मा गर्छ, अर्कोले S phase मा act गर्छ. So most of the drugs target. So if you are using one drug, it will target only a specific part of the cell cycle. But if you use multi drugs, it will target the multiple aspects of this cell cycle. So that's why we use this multiple drugs for the chemotherapy purpose, because it is effective against tumor type. It has different mechanism of action. It acts on different part of the cell cycle, and it is not susceptible to the same mechanism. What is a mutation or causes an efflux pump. So and it has some different side effect profile as well. So that's why the doctor often prefer this combination chemotherapy to this cancer patient. Now coming to this immunotherapy, we have already seen the cytotherapy or cytotoxic therapy, means giving drugs and killing the cancer cells. What here we do is we make our immune system strong. Why? In order to kill the cancer cells. So what are the different ways we can make our immune system strong? Means we can give the cancer patient this monoclonal antibodies, or we can give patient T cell, or we can give cancer patient a vaccine that is called cancer vaccine to increase its immunity. Hence, the patient is strong and can fight against those cancer cells. So here you can see these are the various monoclonal antibodies, which I have already discussed in previous seminar. I'll not go much more detail about this. So these are the various drugs: rituximab, trastuzumab, cetuximab. These are the monoclonal antibodies. What they do is, here you can see. So these are this is the cancer cell. This is the breast cancer cell. So it has one receptor. You can see here. This is HER2. HER2 receptor is there. So what we do is like we give drugs such as trastuzumab. So this is our trastuzumab, means monoclonal antibody. What it does, like this monoclonal antibody will attack this receptor, HER2 receptor. Here you can see, here is an antibody receptor complex. Antibody antigen receptor complex. So now this will signal other immune cells come and attack this cancer cell, and hence it will help in patient get rid of this kind of cancer cell in their body. So this is the principle behind this monoclonal antibodies immunotherapy. And second is the T cell therapy, means we can give patient a T cell so to fight against the cancer. So the most popular is the CAR T cell. So here this CAR and T, the CAR it stands for chimeric, A for antigen, and R for receptor T cell therapy. So what exactly this CAR T? It's a T cell that is not generated in our body. It is modified and laboratory produce, means we can produce it in the lab. So first produce T cell in the lab, and then when we have enough number of this modified T cell, then inject this T cell to the patient body. And जब यो इन्जेक्ट गरिसकेको हुन्छ यसले स्पेशल रिसेप्टर हुन्छ, द्याट इज कल्ड chimeric antigen receptor. So different types of cancer have different antigens, and each kind of CAR T cell therapy is to fight a specific kind of cancer antigen. So this technique of this CAR T cells, it involves changing a person's immune cells to recognize and fight off cancer cells inside the body. So here is the flow chart to show how we can prepare this this T cell. So पहिला के गरेछ भनेपछि this is the patient को blood बाट we isolate the T cell. त्यसपछि यो T cell लाई चाहिँ हामीले ल्याबमा चाहिँ एउटा RDT recombinant DNA technology बाट चाहिँ certain T cell को gene लाई चाहिँ हामीले modify गर्ने त्यहाँनिर. So we use this technology CRISPR to add a new gene to this T cell. यो पेशेन्ट बाट निकालेको T cell मा चाहिँ थोरै मोडिफाई गर्ने with the help of CRISPR to add a new gene. New gene ले के गर्छ त new receptor लाई चाहिँ कोड गर्छ. So new gene adds a special receptor that is called chimeric antigen receptor to T cells and make the CAR T cell. त्यसपछि हामीले यहाँ बनाइसकेको छ. So यो हामीले चाहिँ these are the modified T cell that is prepared in the lab. त्यसपछि हामीले यो पेशेन्ट लाई चाहिँ फेरि इन्फ्युज गर्दिने अनि सुकेरले के गर्छ पेशेन्ट को इम्युन सिस्टमलाई चाहिँ इन्हान्स गरिदिन्छ to fight against that cancer cell. Now third treatment is the vaccine. So we are very much familiar with this vaccine. यसले के गर्छ नि भ्याक्सिनले चाहिँ हामी चाहिँ भ्याक्सिन भनेको एउटा फरेन एन्टिजनलाई चाहिँ बडीमा हाल्दिने, त्यसपछि बडी विल प्रोड्युस इम्युन रेस्पोन्स against that foreign antigen to produce antibodies. मतलब एन्टिजनलाई बडीमा हालिसकेपछि बडीले एन्टिबodies बनाउँछ to fight a disease. So this is the principle behind this vaccine. Coming to this cancer treatment, यसमा चाहिँ के भन्दाखेरि चाहिँ यो क्यान्सर भ्याक्सिन ट्रिटमेन्टमा चाहिँ this is try to get the immune system to attack cancer cells that are already in the body. So यो normal vaccine र cancer vaccine मा चाहिँ cancer vaccine ले चाहिँ खाली cancer cells लाई मात्र attack गर्छ. So this cancer vaccine can be made. कसरी बनाइन्छ भन्दाखेरि they are made from the cancer cells or part of the cancer cells. So a patient has been diagnosed with cancer, तो cancer cell को whole लाई लिने or as a कुनै एउटा part लाई लिने, and we treat as an antigen. So त्यो cancer cell as antigen लिसकेपछि त्यो antigen ले चाहिँ हामीले फेरि patient लाई infuse गरिसकेपछि the vaccines are made to recognize these proteins on these cancer T cells, and this helps the immune system to recognize and attack those cancer cells. So hence, the vaccine might eventually help to stop further growth of the cancer, prevent the cancer from coming back and destroy cancer cells left behind other treatment. And similarly, recently, Russia 2025 मा चाहिँ आफ्नो general public लाई चाहिँ भ्याक्सिन जुन 25 देखि दिन्छ यो चाहिँ mRNA भ्याक्सिन. त्यसमा चाहिँ के गरिन्छ भनेपछि रसियाको रिसेन्टली जुन अहिले हाइली डिबेटेड भ्याक्सिन छ त्यसमा चाहिँ क्यान्सर सेलबाट चाहिँ डक्टरले के गरेछ भने जो पेशेन्ट अलरेडी जसलाई क्यान्सर छ त्यो पेशेन्टबाट चाहिँ डक्टरले चाहिँ के गर्छन् चाहिँ क्यान्सर सेलको बाट तिनीहरूले mRNA आइसोलेट गरेछ. त्यसपछि त्यसलाई त्यो आइसोलेटेड mRNA लाई चाहिँ एआई ट्रिटमेन्टबाट चाहिँ उनीहरूले चाहिँ भ्याक्सिन बनाएर फेरि त्यही पेशेन्ट जसकोबाट हामीले यो mRNA निकालेको थियो त्यही पेशेन्टलाई चाहिँ इन्फ्युज गर्ने भनेर चाहिँ रसियाले चाहिँ भनेको छ. So the cancer vaccine can be the following treatment. The following cancer treatment vaccines are most commonly studied, whereas whole tumor vaccine, antigen vaccine, dendritic cell vaccine, and DNA vaccine. And recently, Russia ले चाहिँ mRNA vaccine बनाउँदैछ यो cancer को लागि चाहिँ. So whole tumor vaccine मा चाहिँ whole cancer cell नै लिने as a antigen रूपमा treat गर्ने हो. अनि यसमा चाहिँ antigen vaccine मा चाहिँ it's made from the antigen in the cancer cell. And dendritic cell vaccines, they are made from the dendritic cells that grow in the lab together with the cancer cells, and whereas these DNA vaccines are made with a bit of DNA from the cancer cells. So cancer vaccine can be divided in two types, preventive or therapeutic. Preventive means it only helps in prevention, whereas in therapeutic already diagnosed भएको केसलाई चाहिँ यो भ्याक्सिन दिइसकेपछि it helps in the treatment of the cancer. So preventive cancer vaccine मा चाहिँ most अहिले अथवा जस्ट मोस्ट कमनली युजमा चाहिँ हाम्रो HPV भ्याक्सिन छ, अर्को चाहिँ हेपाटाइटिस बी भ्याक्सिन. सो दिस भ्याक्सिन आर युज फर प्रोफाइलेक्टिक पर्पज टु प्रिभेन्ट पेशेन्ट टु डेभलप क्यान्सर. for HBV, let's say cervical cancer, whereas hepatitis B virus, let's say for liver cancer. And therapeutic cancer, let's say to treat, the patient has already diagnosed with the cancer, this patient has therapeutic vaccine to make the patient cancer free. For example, FDA approved vaccine or prostate cancer vaccine approved or bladder cancer vaccine. So this prostate cancer vaccine, bladder cancer vaccine is given to those patient who has been already diagnosed with this prostate cancer, bladder cancer respectively. Another one is the melanoma vaccine. And coming to this, there are some other anti-carcinogenic substances that is commonly occurring in the nature. Means there are some substances in nature which we consume. It has properties of anti-carcinogenic. For example, here you can see beta carotene, vitamin A, vitamin C, vitamin E, and lycopene. So these are naturally found. These compounds, they have anti-carcinogenic, antioxidant. And there are some other anti-carcinogenic compounds. For example, here you can see in this diagram, one is quercetin and other is this cinegreen. Quercetin, it is found in the broccoli, this is red fruits and onions, whereas the cinegreen, it is found in broccoli, cabbage. So these compounds, and also in this mustard seed. So these compounds has ability to fight against the cancer. So these are some naturally occurring anti-carcinogenic substances. So these are the references for this this webinar. With this, I would like to thank you, and I would like to hand over to Dr. Srijana ma'am. Thank you so much.
Thank you so much, everybody. For so long, all of you have stayed and tried to gain the knowledge about such a vast topic of cancer. We have stayed for 4 hours for this study of cancer, but still it is not enough. Still so much things are still there to know about. Then also, now I would like to open the floor for discussion. Any questions regarding the presentations? Even though it's late, one or two questions we can have.
From the students, if there are any confusion, you can ask student only.
I know डिपार्टमेन्टमा बसिरहेकोहरूलाई एकदम गाह्रो भइसक्यो होला. लम्साल सर हुनुहुन्छ. लम्साल सर, Dr. Bizia, anything you'd like to add upon?
No, addition भन्दा नि, it was a good topic, elaborative थियो. बिचमा I kind of like I was writing सुरुमा सबै making my own, but when you know Snehaको part आएपछि I felt like lost एकछिन. But again, nice effort from everyone. Srijana ma'am, thank you for taking the initiative. Something she did it. She wanted you all as a student to experience, how to conduct a webinar, how to, present online. You all must have recognized now the challenges, data pack handy राख्ने देखि लिएर, how to at least handle the technical issues, how to be ready. I hope you all have like at least केही थाहा छैन जुमको बारेमा अथवा अनलाइन वेबिनार कसरी गर्छ भन्नुभन्दा पनि this was a good experience for all of you, I believe, and thank you to Srijana ma'am, because it takes a lot of effort. The moderator topic, but to think, to plan, to moderate she took a very huge task. So thank you, ma'am, and I hope all of you have been benefited. Sabale compile garo, slide like, keep it together and share it. And in between, you must have lost track of the presentation, so you do have the recording, so you can take the recording like you can upload it and in your YouTube channel, and you can share the link. If you want to make it public, you can make it public. If you don't want to make it public, at least jojoलाई link share गर्यो उसले मात्र open गर्न मिल्ने option राख्यो भने all of you can go and revise anytime. So I think विशालले त्यो responsibility लिऊ. If you feel कि होइन बिचमा lot of distraction छ भने don't make it public and just share the link and जोसँग link छ उसले चाहिँ open गर्न मिल्ने गरेर upload गरिदियो so that you all can revise it for future as well. Right? Share it in the group. And thank you everyone for your patience, and we hope that it was beneficial to all of you in terms of acquiring some knowledge at least. A question सोधेर तिमीहरूलाई टेस्ट गर्नु भनेको it's like giving you a torture at 6 p.m. So I won't be doing that right now. I have a few questions noted down, but we can discuss that later sometime when we are done with the recording, watching the recording, and we can say, okay, we'll have the discussion revision of this webinar भनेर we can do it some other day. Thank you ma'am, thank you everyone. Lamsal sir, हजुर.
Thank you so much for the organizers, Dr. Vijay and Srijana ma'am.
Sir, sabai Srijana ma'am took the initiative.
So, and I thank again for considering my request that I'll be attending this seminar, and for the 4 hours continuous I'm sitting here, bearing with the pain, and I congratulate all the speakers that they did their best in bringing out the online presentations to its international form, I think I should say, because in most of the time, even in the international arena also, there are many technical hiccups there, many things emerges, but I think I consider this one, it has gone smoothly, bearing some problems that is not in our control. Okay. Anyway, there are many questions which has raised, although it is a big topic. It is a big topic, taking almost 4 hours continuously sitting in one place. It makes it very difficult for the attendee also, not only for the presenter but for the attendee also. You don't want to miss. I didn't want to miss any portion of it. I didn't do it. I had my tea. I had my snacks also. That is the benefit with this topic. But since there are many things which are upcoming, I request everyone to file it up and then make it in a brochure type of copy that should be submitted to the department. Then we will be discussing. And this is the way that small topic will come. Then that can be added. And then we can also participate in the as a health worker in the treatment of our in the fight against the cancer also. So I congratulate everyone, and I'm always saying that you should be part of the focal team, which should be participating in one particular type of treatment or diagnosis and all, because your area should not be just restricted to the diagnosis. You should be the part of treatment or part of preventions. Okay, then only it will be broadened. So I don't have any doubt that you cannot do it. And I'm always encouraging that you should participate. And this is how it is coming. Congratulations again to everyone, and best wishes for the further seminars. Next time, I request that let the seminar be not more than 2 hours, so that we can make it for 2 days. So we can keep half an hour for the discussion, or like that, 1.5 hours for the seminar, half hour for the discussions. And if it is not continued, next day we can do it. So this way, most of the topics can be covered, and everybody will participate. Thank you so much again for the presentations. We'll be discussing in the department. Thank you.
Thank you so much, sir. If you would not have agreed to the this kind of seminar or webinar, rather, we would not have done this today. Thank you for allowing this form of the presentation for seminar. And I would like to thank all the presenters for their elaborate presentation and their in their respective topics. And I hope all our PG students must have understood and learned from each other today in this 4 hours. It was such a long time and big topic which you have discussed with each other. I would like to congratulate all of you for doing such a nice presentations. And I would like to thank Dr. Bizia for helping us so much, cooperating and providing us Zoom link for so long hours. This, if it would not have been her, we could not have been doing this presentation today.
Thank you.
And it was like I feel like the cancer is such a vast topic. We could not have dealt anything in piecemeal. Okay, it was like I felt like buying a piece of cloth, cutting it down and stitching it to make a new cloth. It felt like that, with so many presenters, 11 presenters presenting in various fields of cancer. And I feel like they were like expert in those fields, and they are learning to be expert also to in those fields. I think later on I hope that they become like that in those fields. And I thank all the participants for such a long time. They have been sitting there. It is not so easy task. Whoever was sitting in the department, it was really hard for them. I think we should have one nice high tea for that. Should have for as a reward for this.
You all can go
It would
and
have been nice today itself, but sometime we can have as a reward. Okay. And I think that's enough.
so we can end the meeting recording. Vishal, once I end the meeting, recording आफै convert हुन्छ तिम्रोमा, it will be auto converted. Don't stop it, because if you stop it, it is going to be lost. So once I end the meeting.
Next time we'll not restrict it only to our own students. We'll try to broadcast outside also. We'll see if it can be possible, so that we'll expose the potentialities of our students, PGs, faculties as well. Thank you.
Thank you
so much, sir.
Thank you. Ma'am, should I end? Then Gauri.
Okay, sure. हुन्छ
Bye
है अब सबैलाई thank you. Good night.
Good night. 1, 2, 3.